Uh, First and foremost, my name is Lotfi and not Dr. Merabet. I think that's the first important thing to remember. Listen, we're all friends, we're all colleagues. This isn't a place for formalities. I want everyone to sort of be comfortable and exchange ideas and so on. So I just want to tell you that that's how I see these things. I think the first important thing to mention. In terms of my background, I'm a clinician scientist. I'm an optometrist. In terms of my clinical training, I work a lot with patients with visual impairment, both of eye causes as brain-based causes. When I was an intern, I spent a lot of time at the Perkins School for the Blind, and as you might imagine, that had a big influence on me in terms of the type of work I wanted to do, not just clinically, but also from a research standpoint. I'm also a neuroscientist. I'm very interested in the brain, and in particular how the brain adapts to visual impairment. Again, both of ocular as well as brain-based causes. Our lab does a lot of work in a variety of arenas, including brain imaging, looking how the brain is wired, how it talks, how it talks to other areas. We also develop a lot of virtual reality techniques to develop ways to assess visual impairment beyond just the classic things that you see in an eye doctor's exam room. And I'll be showing you some of those examples actually in the second half of the presentation today. And then the last thing I'll tell you about myself is I have a background in public health, which I consider is extremely important because I feel that the research that we do should not only translate in terms of the clinical care of these individuals, but ultimately into the classroom and into the real world. It's a tough line to cross, or I should say a tough line to walk, where on one side you have to do really basic fundamental research, and on the other side you have pressing questions that teachers and parents and patients ask you. But hopefully we'll continue. I really think having these sort of dialogues and staying connected to the community are extremely important because it helps us guide some of the questions that we should be answering in the lab as well. So again, I learn tremendously from these opportunities. In terms of today's presentation, I'm going to break it down into two sections. Hopefully you all have the notes with you. The first hour is going to be just an introduction to the brain and visual system. It may very well be a review for many of you, especially when it comes to the brain anatomy and so on. But I've tried to highlight things that I think are particularly relevant when it comes from a developmental standpoint as it relates to the developing brain, the response of the brain to injury, for example. So I'm going to really try to spend more time on those aspects than the particular anatomy per se, which I'm sure you're all very familiar with. We'll take a break for about 10-15 minutes, stretch our legs. And then the second hour will be concentrating more on neuroplasticity and in particular CVI. Neuroplasticity in the sense of how the brain changes. And CVI is really an interesting situation. Obviously, we all know that CVI is an important condition, but that we should be paying attention to. But also from a neuroscience standpoint, I hope to convince you that it's a really interesting model, for lack of a better term, to understand how the brain adapts to tremendous pressures in terms of an injury, for example. So that would be the idea for the second hour. In terms of moving forward, it's a bit of a challenge. What I've put together here is, I guess, a teaser, if you will, of a course that I typically give at the Perkins School for the Blind, which is 4 to 5 hours. Actually, we'll send you the link if you're at all interested in attending the entire course. I see a heart coming up there. Thank you. And the idea is really just to give you, again, sort of a sample of the material that we cover. It's only going to be 2 hours, like I said, of otherwise a 4 to 5 hour course. Unfortunately, there's a lot of presentations and demos that I have to be in person in order to do it. So I've only had to, I really just concentrated on things, like I said, that I hope are pertinent for you to get started. And of course, we can certainly discuss further, growing the course and continuing if you think this is helpful. And then the last thing I mentioned before we start, this is pretty dense material, and I realize there are many of you on there. I know many of you are teachers for the visually impaired, rehab specialists, orientation mobility experts as well. We've even had parents jump in on these courses in the past as well. Just in the interest of keeping the flow and staying on time, the only thing that I would ask is that if you have a question during the presentation, really specific to the content. So for example, I went too quickly for our friends, for example, who are visually impaired. prepared and maybe not catching some of the visuals that I'm using, if they want a greater description or more time spent on that describing, I'm happy to do that. So Ashley is agreed to look at whoever is raising their hand or is in the chat, and I'm happy to stop and spend more time. But if it's a question more like, oh, I know a kid that has this, or I know, this sort of thing, can we please hold those sort of more general questions to the end of the session, just simply because, like I said, I've done this many, many times before, and those questions tend to derail, the entire, rhythm of the presentation. I'm certainly happy to talk about it, but let's keep them for the general session, or offline. I'm happy to talk with any of you as well. Is that all right? Excellent. I see nodding, so I'm going to assume that's okay. All right, first part, brain organization and function. And here is my first slide, just some opening thoughts from the perspective of a neuroscientist that I think would be useful for our conversation today. And the first line is this: the mind is what the brain does. And that, in a nutshell, I think is a really, really important concept to think about when we talk about neuroscience in general. That's to say, every memory you've had, every decision you make, whether or not you like this presentation, what are you going to do tomorrow? What did you do today? All that is the product of all these brain cells in your brain, chemicals sloshing around in about a 3.5-pound mass. And the idea is that little voice in your mind, that little decision maker in your mind, is the product of all those actions that's happening inside your brain. As William James, a very famous philosopher and psychologist, said, he goes, the mind is not a thing, it's a process. And it's really interesting I think it's important to kind of distinguish those things. The mind is the product of all these interactions and activity that's going on inside the brain. And just from a historical account, I think is kind of interesting. In the early days of St. Valentine, he was able to go around with these little sermons of about 1 or 2 minutes. And when people who had seizures, for example, collapsed at that time, we thought they were somehow possessed, from demons and so on. And he had these nice little 1.5-minute seizures. And sure enough, these patients came through afterwards. And he used these it was believed that his sermons somehow calmed the individual down. St. Paul, and then just by no means am I trying to make any blasphemous statements or anything, but it's just interesting to kind of look through history. St. Paul was known to be an individual with very, very vivid hallucinations and visions of the future and so on. And we know, and there's some interesting speculation, that maybe St. Paul had something called temporal lobe epilepsy, where the temporal lobe, we know, is the part of the brain that stores visual memories. And epilepsy is a brain disorder that creates this high level of activity in that area. So maybe what St. Paul was describing were these images that he had in his mind due to really a disorder of the brain. The rat pleasure center, this was a very interesting study. There's a particular part in the thalamus of the brain that if you send a small amount of electrical current and you have it wired to a little lever like this, the rat will sit there and just auto-stimulate for hours on end, even if it hasn't eaten in a day. There could be a mate, who's ready for mating next door. The rat will completely ignore it just because this auto-stimulation, for example, is so powerful for them. So they call this the rat pleasure center. There is also this interesting work called the remote control bull. There was this individual, this Spaniard, who put this electrical device inside the motor cortex of a bull, and as the bull was charging, he would not detonate it, but use an electrical charger, which would send a small amount of current to the motor cortex and would make the bull freeze. And as you might imagine, coupled with a cape and so on, would make for an interesting spectacle. Michael Persinger invented this thing called a God helmet about 15 years ago, where particular, again, stimulation along the temporal cortex would create these sensations in individuals who wear the helmet of incredible divinity and sense of the presence of God and so on. And again, I'm just trying to say here that things that historically were really esoteric and things that we couldn't understand are now more and more entering the domain of neuroscience, and we're closing this gap, as I said, between the brain and the mind. By understanding how the brain works, we have a better understanding of how the mind works and ultimately who we are. So that's what I meant by that introduction. When I was an intern, I had a mentor, a really, really good mentor in neurology, and he said, the most important thing you need to remember is this: neurology is like real estate. The three most important things are location, location, and location. And of course, what he was talking about is that if you look at the surface of the brain, the cortex, it looks pretty much all the same, right? There's nothing there that would indicate to you that any part of the brain is different in any way. But yet we know today that different parts of the brain do different things. There are specialized areas for vision, which you know is the back of the brain. The front of the brain we know is responsible for working memory. There is motor cortex, sensory motor cortex, the cerebellum responsible for coordination, parts for hearing, parts for taste, speech, spatial awareness. And it's this localization of brain function that's extremely important, because the idea is that the two are tied together. If we know where the lesion is in the brain, we should know what type of deficits that patient has. Conversely, if we see particular deficits in behaviors of a certain individual, we should be able to hypothesize where the damage is in the brain. So that brain structure connection, or that functional structure connection, is really an important concept in neurology and neuroscience in general. We always go back to that. If we know things, where things happen, we can figure out what the deficits are. And if we know what the deficits are, we should have a good idea of where the damage is in the brain. Just going a little bit further here, showing you some of the main sort of subdivisions or ways to think about the brain. The most obvious is to think about the two hemispheres of the brain that you saw there, the left and right. Deep into the brain, what we call the diencephalon, this is the thalamus. This is the gateway, the entry of information into the brain. And don't worry, I'll spend more time on these structures. And this third part that you see here is called the corpus callosum, which literally means big body. And that is the connections between the two hemispheres. So the takeaway message here is you have a left and a right hemisphere. You have deep structures into the brain where information is coming in and going up into the cortex. And you have this corpus callosum, which allows communication between the two hemispheres there. So the other thing I want to point out here that I think is very, very important is indeed the cortex is the outer layer of the brain. And again, we'll spend more time talking about that. We also know the ventricles, these dark areas in between that are always showing a black, for example, on MRIs and so on. This is fluid, right? There's no neural activity happening in these areas. This is fluid that we know is important for nourishing the brain and also allowing for toxins to be removed or to flush out. You can think of it almost like the sewage system, if you will, of the brain. And it also helps the brain to absorb shock. It's almost like shock absorbers. So when there's a trauma to the brain, it isn't a rigid body. It can compress into the fluid and kind of rebound back. So the fluid inside the brain is extremely important to protect it as well. As I mentioned, there are subcortical structures, namely the thalamus. This is where information enters the brain. And the important thing to take away from this figure is in between the thalamus and cortex, or gray matter, is what we call white matter. White matter is the communication cables. This is how the thalamus communicates with the cortex and vice versa. And it's also how the cortex communicates with itself. So it's very, very important to realize that there's three levels of the brain. And when we think about the brain, we always take these three levels into consideration. And again, we'll spend more time on this. So deep subcortical structures like the thalamus, where information is coming in. White matter, which is how information is exchanged. It's how the brain talks to itself. Gray matter, the cortex around, which we know is where the specialty work is done. It's the seat of conscious thought. It's where processing largely happens. So just a quick sort of overview of the organization. We're going to spend a little bit more time sort of breaking this system down for you. So the thalamus, the entry point or the gateway to the cortex. All senses, vision, hearing, taste, touch, and proprioception, which is our ability to know where we are in space, how our limbs and hands and so on are oriented in space, all pass through the thalamus. As you see here, for example, let's, in the interest of the eye, here is the eye. You see this connection going into the thalamus deep inside the brain and then sending information to the visual cortex. From sound, the ear sends information to the thalamus and then sends information to the auditory cortex. And this is a recurrent plan or a recurrent strategy. Information from outside world enters the thalamus and then enters the cortex, right? So the thalamus is the gateway of sensory information to the rest of the brain, right? There is one sense that doesn't follow this model, and that is smell, right? If you notice from the nose, sensory receptors go straight to the cortex. It doesn't go through the thalamus. And that's kind of an interesting thing. The theory is smell or olfaction is one of the oldest senses from a developmental standpoint. And it has to do with this theory that because the first early mammals were very, very close to the ground, or the idea was that smell was one of the most predominant senses that these animals used. And as mammals developed and as other organisms developed higher and so on, the other senses started becoming more prevalent. So it could be a reflection in terms of overall development of the brain. But again, the take-home message here is the thalamus is the entry point of sensory information. And we'll get to why this is important in a few minutes. The second level that I want to mention is what's called the limbic system. The limbic is kind of what we call the paleo-mammalian. It's the old brain, if you will, before the fully developed cortex. And in the limbic system, we have three major parts: the hypothalamus, the amygdala, and the hippocampus. What's important to take away from this is the part of the brain that we know is very, very important for emotion. Right? Emotion processing, aggression. It's also important for memory as well. It plays a very, very important role. But think of the limbic system as the emotional brain, right? That is the part that really kind of gives us polarity to things like hungry, thirsty, angry, sad, attack, flee, all those sort of things. So very, very primitive behaviors, if you will, are attached to the limbic system. And I'll get back again why I think this is important. Here's just a nice example. This is an fMRI study. fMRI is a technique that allows us to see which part of the brain is particularly active during a particular task. This is the limbic system here activated in an individual with, quote, neurotypical development, right? So you can see the limbic cortex here on the side and in the middle. This is the limbic system really lighting up quite a bit in an individual with neurotypical development. Here's another individual. And if you notice here, there's almost no activity at all, just a smidgen of activity. So you might ask yourself, why would this particular person not have any limbic cortex activity? Anybody want to throw it an answer, what they think's wrong with this person? If you haven't already looked at the notes. It's a psychopath, right? A psychopath clinically is someone who does not have strong emotional valence, does not have strong emotional activity. And it's interesting, as I said, here's a perfect example of how the behavior fits with the location and the activation of the brain, right? So a very just simple example of how the two should fit together. If we don't see strong activity in the limbic system, we should anticipate emotional related behaviors or emotional related issues, right? And again, just a nice example how the two fit together there. The third and the last piece in our conversation is the cortex. And this, again, probably something you're all very, very familiar with. The cortex is the seat of high-level sensory processing, thought, and reasoning, right? So the thalamus, entry point of information, the limbic system, our emotions, the cortex, really higher, or the executives, or the high-level processing of sensory information and thought, right? Useful to separate this into four lobes, which we'll do here. The frontal lobe, by name, is the front of the brain. This, interestingly enough, is the last part of the cortex to develop, right? The rest of the cortex tends to develop, and the frontal cortex typically is the last thing to develop. Interestingly, full development is about the age of 13 and 14. Any of you who have raised an adolescent knows exactly what I'm talking about. Right? Again, fits with the development. The frontal cortex is typically the last part of the brain to develop around 13, 14. And what do we notice? This radical chain in teens, right? They have this sort of almost risk-taking behavior, not the greatest decision-making. And as that frontal cortex develops, we know that hopefully things start falling in line. So again, fitting, structure, and function, again, very, very close. The parietal cortex up top here, we know that this is the part of the brain responsible for spatial processing. The temporal cortex along the side here, we know very important for visual memory, some, again, a strong sort of visual component and memories in that area, as well as certain language areas. And finally, the occipital cortex, the back of the brain, largely responsible for visual processing. And the takeaway message from here is think about all the things that the brain has to do, all the senses, memory, motor planning, et cetera, et cetera. Look how much is dedicated to vision. It's a huge chunk of real estate in the grand scheme of things. About 30 to 35% are some typical estimates that the cortex, the visual cortex, is about 30 to 35% of overall cortex that's dedicated just to processing visual information. So quite striking. Here's just a review of what I've just mentioned. Frontal cortex involved with executive functions, creating associations and planning. There's certain speech areas as well, focusing attention. So again, all these are what's called executive functions, decision making. Temporal cortex on the side, parietal cortex, I should say, second. Spatial processing, knowing where you are in space, where things are in space. We also know there's some areas involved with language comprehension as well as calculation as well. People who are very good in mathematics, we know typically have certain areas that are quite thick in the parietal cortex. We also know that people who get damage to the parietal cortex have problems localizing things in space, what we call neglect syndrome, for example. Temporal cortex along the side, very important, as I said, with hearing, memory, smell, higher order visual areas. Some of you probably know that the ventral stream, the part of the brain responsible for knowing what objects are, or our ability to recall the names of things and identify objects, lie in the temporal stream. And then finally, of course, the occipital cortex, which is involved with early visual processing. And again, I just want to highlight the fact to think of all these functions that the brain has to do. Notice how much real estate is dedicated to the process of visual information. So let me kind of put all this together. I realize probably coming very quickly at you. Let me kind of give you a sort of a model of putting things together. As I mentioned, the gray matter is the outside cortex, right? The white matter is the cabling, is the information superhighway, if you will, of how information connects within the brain, cortex to cortex, as well as cortex to thalamus. The reason why we call it gray versus white matter has to do with anatomy. Gray is because this is where the cell bodies are of the brain cells inside the brain. The cell bodies that are responsible for capturing information typically are here, these large little bodies here, as you see, right? Then they all have this long piece that's called an axon. An axon is how a brain cell sends its information and communicates to another brain cell. And we know that these axons have something called myelin. Myelin is insulation around the axon that allows the information to be propagated and sent very, very quickly. And that myelination tends to give the cell a bit of a white color. And that is the differentiation between gray matter and white matter, or cortex and white matter. It has to do with the myelination, right? As the other thing I want to mention, information sent to the thalamus and up to the cortex. The other thing that's important to realize is that this isn't a one-way street. The cortex also sends information back to the thalamus, right? So there's a constant exchange of information. I think that's an important thing to keep in mind. Sensory information enters the thalamus and then enters the cortex, and then the cortex sends information back to the thalamus, starting this iterative process, this loop, this feedback loop. And that's kind of how we perceive the world. It's kind of an underlying principle, regardless of the sense that you're working with. Information comes in, it is analyzed, it is sent back to the thalamus to compare with new information that's coming in, and the loop continues on and on, right? So that's an overall organizational principle, if you will, of the brain. Here's an analogy. I hope this is helpful. You may have heard this before if you've seen me lecture. I use this analogy, which I hope is helpful. Think of the brain as a high-rise executive building, right? And let's break this down, kind of makes sense. I call this the building analogy. The cortex is like the cubicles on the high floors. This is where the executives sit. These are the bean counters. These are where the decisions are made. These are the specialists. These are the guys with the high paying salaries, for example. These are the guys, these are the people who are making the big decisions, right? The white matter is kind of like the elevator system or the stairwell system. That's how things are shuttled from floor to floor. That's how information gets from one floor to another, right? And the thalamus is kind of like the lobby. That's where information comes in, and that's where information comes out, right? Or information leaves, I should say, right? I think this is a useful analogy because you can now speculate what happens when you have damage into these particular areas. If you, for example, knock out a bunch of cubicles, if you have cross-training, if the people, the cubicles next to you know how to do that job, you can still keep the job going. And we know this is true, for example, if a patient has a stroke and the stroke isn't too extensive, if the brain areas next door are kind of cross-trained, they can pick up that function and be the source of that recovery, right? In the case of white matter, if we have damage to the elevator system, well, maybe there's other pathways that people can use the stairs, for example, to get up with. So in the case of recovering from injury or brain damage, if there's extra pathways to get to those same areas, maybe we can, again, recover function. The last thing I'll mention is the thalamus. If the thalamus is destroyed, or if the lobby is destroyed, then nobody gets in and then nobody gets out. And that is actually kind of what we see in terms of brain damage as well. We know that early damage, extensive damage to the thalamus, really tends to have a really bad prognosis, typically, for a child in terms of development. Why? Because that information isn't coming in and it's not leaving the brain either. It doesn't matter how good an elevator system you have or how good the executives have. It all kind of starts at the level of the thalamus. So hopefully that's a useful analogy to kind of understand and parse the different parts of the brain. Yeah, I'm going to take just a quick second here, Ashley. Any thoughts? Any questions? Happy to repeat anything. Are you seeing anything appearing?
There's nothing in the chat so far.
Okay. All right. So either this is very clear or incredibly boring. I will.
No, I think it's just kind of maybe, you know.
It's overwhelming. It's quite a bit.
I think it's great.
Okay.
Carry on. Yeah. The building is a great analogy.
Okay, good. Listen, for what it's worth, don't be too concerned. This is, I'm giving you in 20 minutes, which should theoretically be a full, graduate level course. I'm just trying to give you some highlights to kind of think about the overall organization. I think things are going to become more and more clear as time goes on, and you're like, oh, okay, that's what he was talking about, and so on. So if you guys are feeling a little bit overwhelmed or this is going too fast, don't worry. It's a little bit by design. Okay.
We're getting some very interesting, very clear. Yes, a lot of information, but clear. So
okay,
carry on.
Good.
Yeah.
All right. Excellent. Let's keep going then. A couple of comments I think that are very, very important. Again, lots of graphs and figures, but I'm going to give you sort of the takeaway messages from this on brain development in general. Here's a very, very important thing to keep in mind. The basic structural and functional framework of the brain is in place by the second year of life. In other words, by the age of 2, you're basically born with all the machinery you're going to have for the rest of your life. And it's all pretty much downhill from there, to be perfectly honest with you. And I'll show you the data. After the age of 2, brain development is mainly reorganization, fine-tuning, plasticity, which we'll talk about, and remodeling of established circuits and networks, right? There is also something called the critical period. You may have heard this in your past courses as well. The critical period is the stage or time period in a developmental in the development of an individual to which the brain is especially sensitive to environmental stimuli. One thing that's important to think about is that this is not a closed window. When the critical period ends, and typically that critical period is between the age of 2 and 7, right? What do we know about the age of 2 and 7? That's a very, very good time, for example, to learn a language, a very, very good time to learn a skill. After that, we certainly can do that, right? But it's much harder, right? So the critical period is that time in brain development where the brain is particularly receptive to environmental stimuli. We know this, for example, in terms of like accents. If a child learns a language within this original period of 2 to 7 years, typically they speak like a native speaker, right? Whereas if you learn a language later on, you can still do it with a lot of effort, but typically that person will speak with an accent, right? So this fine period, this age between 2 and 7 years, we call the critical period, is quite important, like I said, in terms of that's when the brain is very, very sensitive to information. But I don't want you to think that it's a closed window. It doesn't mean that nothing happens after that. It just means that things are a little bit harder to kind of coax the brain into change. So let's look at this graphically. On the y-axis here, I have percentage change. The higher the number, the more a certain variable is changing. On this axis here, I have age. So 20 weeks gestation to birth, 1 year, 2 year, all the way up to 20 years. And as just to reiterate my earlier point, you are essentially born with all the machinery that you have up to the age of 2. Right? So if you look at this red area here, do you see how everything's kind of just, these lines are all kind of exploding? Those first 2 years of development is really an explosive period in terms of the brain. The brain is just doubling cells, forming connections and so on. Then after the age of 2, as I said, that critical period, things tend to level off. Gray matter, that's what I talked about, the cortex, that's the overall volume, tends to be pretty flat. Surface area, how much area the cortex of the brain occupies, tends to be pretty flat. FA, or fractional anisotropy, that's a measurement of myelination, also tends to be flat. Cortical thickness tends to be flat. What do you notice? The only thing that's really changing after the age of 2, or keeps changing after the age of 2, is brain connections, right? And that's an important thing to think about. As we leave this critical period, as we enter this critical period and leave this critical period, the game is about connections, right? So up to the age of 2, it's a numbers game. After the age of 2, it's a connection game, right? And that's actually kind of interesting, because what do we know at 2 years? The terrible twos, right? This is when kids are having all sorts of issues. They have an identity crisis, emotional issues, breaking out into tantrums. Why? Because the brain is trying to connect it all. It's my emotional brain, information coming in. How do I analyze that sensory information? How do I act on it? This is when the brain anatomically is trying to form those connections. So it's not entirely surprising that a child is particularly turbulent at the age of 2, because the brain is switching strategies in terms of how it's getting wired. All right. So hopefully what is probably sort of a lot of data kind of makes intuitive sense to you in terms of the timing, right? So again, just to repeat, the first 2 years of brain development is a numbers game. After the age of 2 up to the age of 7 is this critical period where the brain is particularly sensitive to environmental stimuli. Then after that, things get a lot harder in terms of inducing change. Doesn't mean it's impossible, but it's a lot harder to do, right? Second point I'll make about this that's very, very important is that critical periods are not uniform for different aspects of brain function. We know that after birth, sensory systems tend to have the first critical period. Vision, hearing, taste, all the senses, those tend to be the first things that come online. After that, things like motor functions and language tend to come into play. And then after that, we have things like higher cognition. As I said, remember the frontal cortex, the last part of the brain to develop, big decision making, executive functions, et cetera, et cetera. So the different parts of the brain don't all come in line at the same time. There are different critical periods for different aspects of brain development. And that's important to think about, because this kind of sets up a domino effect. If there is an injury somehow, blindness, deafness, perhaps an individual who loses a limb early on in development, it tends to have a domino effect for all the other things that need to develop, right? So it's important to think about that there is timing matters in this issue, right? For example, if sensory function is intact, but we have an impairment in language, maybe that doesn't mean that then the sensory functions are now impaired, because they've already developed, right? So the sequence is extremely important to think about where you are. And a nice quote from Takao Henshi says, windows of opportunity, meaning these critical periods, are also windows of potential vulnerability as well. So it's a knife that kind of cuts both ways. So timing is extremely important. And I want to drive that point home in a couple of slides as well. Here's a great example of exactly what I mean in terms of the visual system. Here's the visual acuity of a newborn, but this has been done through various simulations and based on empirical data that we've collected in the scientific community. From a newborn to 4 weeks to 8 weeks to a 3-month-old, all the way to 6-month-old, there is dramatic change in terms of the overall acuity of the visual system, right? By 6 months, it's essentially at an adult form, right? So from newborn all the way to 6 months, huge change, right? In terms of overall performance. But from 6 months to an adult, decades, it's really just fine tuning, right? The jump from 6 months to adult is not anywhere as dramatic from newborn to 6 months. Everybody see that? Yeah. So the important thing to think about is those early months, those early years are explosive. That's where a lot of change is happening. And that's why the timing is so critical, because this is when the brain is really, really active. Like I said, playing the numbers game. After that, things become more of an issue of fine tuning. All right. So nice example of how visual acuity seems to follow this model. Other things to think about when it comes to how to build a brain, again, to reiterate this point about numbers versus fine tuning. In the early stages, as I said, it's kind of like building with Lego. You're putting all the blocks together, right? You're adding, you're adding, you're adding, you're adding, you're building, you're building, and you're building. As we move into the critical period and into adulthood, it becomes more like pruning, right? You've got this big structure, and you're really just kind of now finalizing those last connections and so on. So as I said, there's a strategy shift in terms of brain development. At the beginning, it's much more of a numbers game, putting all the pieces together. At the end, later in life, after the critical period and into adulthood, it's fine tuning, it's pruning. You've got a solid base, you've got the solid branches, and really now what you're doing is kind of polishing and making those connections a little bit more obvious, right? So again, there is a strategy shift that we know fits with a child development and overall development as well. There is also something, like I said, that timing matters. And I really want to drive this point home, because very often, for example, we know that if a child loses their vision late in life, they're very, very different than a child who was born with visual impairment. And the question is why? Well, you have to look at it from a timing standpoint. And the way that I try to explain this is using what's called a clock analogy. So think of a mechanical clock, right? If I have that clock and it's keeping time and I drop it, right, it's going to have an error in time, right? In that particular case, if I have a situation where the clock is built correctly and then later it takes a hit, like I dropped the clock, the error in time has to do with how those remaining pieces still function, right? And the error also reflects which pieces were broken as well. So I have a fully assembled clock, I drop it, it has an error in time. Theoretically, if I look at which gears are broken and which pieces are broken, then I should be able to predict what the error is in time, much like our brain situation. The case, what happens when a child is born with a visual impairment is a very, very different situation, because it's a clock that hasn't been built yet, right? That error is happening early while that clock is coming together. And the idea is the error that it shows is a reflection more of the pieces that are there, and those pieces are trying to find each other, trying to still kind of connect. So I want to make you realize that, because what this means is that the strategies that we use, the strategies that we use in the case of late blindness or late onset sensory deprivation may not necessarily apply to a situation of early onset visual impairment or sensory deprivation, because the systems aren't quite the same, right? In this particular case, the clock has been assembled and there are pieces that are broken. In this particular case, the clock hasn't been built yet. So we cannot anticipate that it's going to react the same way to being fixed, so to speak. Does that make sense? So again, timing is extremely important. So just to review this, when we talk about development and recovery from brain injury, the nature of the injury matters. Did it happen early in life? Did it happen late in life? The severity of the injury, the extent, how much of the brain did it affect? The location, as I said, location tells us something. So the nature of the injury is extremely important to take into consideration. Associated factors like the developmental stage, cognitive capacity, and reserve. As I told you, the critical periods are dependent on what the actual development process is. Sensory structures are much more earlier than motor and language, which are in turn earlier than executive functions. So where that individual is in their developmental stage may have different results in terms of the same sort of hit to the brain, if you will. Also, in terms of cognitive capacity and reserve, is how well can that individual adapt or compensate for that particular sensory impairment as well, is an important question, which you all know. And finally, and I think something that is extremely important and very, very much, at least in the neuroscience realm, really not studied as well, is environmental issues like support mechanisms, access to rehabilitation, and so on. This is something that's funny. I'll just tell you a side story. When I work with ophthalmology and optometry residents, and we're in the low vision clinic, very often we play sort of a game where we look at the charts and we see, okay, based on the visual acuity and visual fields and this and that, who do you think is going to have a very good outcome from a rehabilitation rehabilitation perspective, and who doesn't? And it's funny, the students will always kind of look at the data, visual acuity, visual fields, et cetera, et cetera. I don't. I look at the family, right? The family probably tells me more how well that individual is going to continue, compensate, and go through their rehabilitation program. Because I believe that if the support mechanisms are there, that tends to trump all the other issues, like I said, of visual acuity, visual field, et cetera, et cetera. So again, something that's probably very intuitive to all of you, but something that is not really well studied from a neuroscience standpoint. We really don't know the beneficial effect of environment and how to factor that in when it comes to overall rehabilitation. So that was just the introduction of overall brain organization and anatomy. I realize that that was probably overwhelming, but again, appreciate that in 20, 30 minutes, you were seeing an overview of something that normally I teach for 2 hours and normally is taught in an overall course. So I'll take again a quick break. Are there any comments about the overall organization of the brain? Happy to address that.
There's nothing in the chat right
Nothing in
now.
the chat? So far, so good? Okay.
Yeah, so far, so good.
I have a feeling things are going to get a little bit more, things are going to get hot as I start moving into the visual impairment and CVI for sure.
Yeah,
Okay.
I think you're right about that.
Okay, very good. But this is sort of a necessary evil. We have to start with the basic in order to grow. Two nice quotes that I think are really relevant to our conversation. This is the first one by Aristotle. He said, "To see is to know what is where by looking." And I think this couldn't be a more succinct statement in terms of trying to understand the concept of to see. Because we know, I'm sure many of you know, that what an object is and where that object is in space is actually handled in different parts of the brain, what we call the what and the where system, and we'll review that in just a couple of minutes. And the last part, looking, to me, means moving the eyes, knowing where to look in space. And that, again, is, in a nutshell, probably the most succinct definition you can get about the visual system. It's knowing what an object is, where it is, and being able to look for it in space as well. Helen Keller, just a little bit more philosophical, said the only thing worse than being blind is having sight and no vision. I think that's a very, very telling statement as well, because what I want to convince you is that seeing is one thing, but vision is a whole bigger level, right? Seeing is the information that enters the brain, but vision is how the brain handles that information and interacts with it. And that's a whole order of complexity higher that is important to distinguish as well. Just because the information is coming in doesn't mean it's being handled, right? So that's an important thing to think about. Again, probably a review for you guys, just to remind you that the left side of, if you can split the visual field in half, we know that the left side is handled by the right side. The right visual field is handled by the left hemisphere. We know that this information crosses. This is true not just for vision. We know it's true also for motor function. My left hand is controlled by my right hemispheric motor cortex and vice versa. This crossing organization is something that's quite typical that we see in overall brain organization as well. So left controlled by the right, right controlled by the left hemisphere. Another thing that's important to realize is that in the middle here is this optic chiasm. The optic chiasm is where information from the two retinas cross and enter the other side. And we're going to get to why that's important in just a second here. So keep that in mind. Some more details about the organization of visual cortex with respect to the visual world that it sees. So I'm showing you here 4 quadrants in 4 colors, red, blue, orange, and green. And let's march down the visual system and see what happens. First thing you'll notice is that the projected on the right side is now in the left. So right, green, and orange is now on the left side, and vice versa. Right? So the two eyes see slightly different images, and you can convince yourself of this if you just look at the screen and close one eye and then close the other eye very, very quickly, go back and forth. You'll notice that the images are very, very similar in principle, but they're kind of jumping left and right. And we call that disparity. That slight difference in the two images, when fused by the brain, gives you the perception of depth, right? So that's an important thing to think about. And that's what I mean by sight versus vision. Information coming into the brain versus how the brain processes that information is a whole order of magnitude much higher, right? So as we enter the visual system, the first thing we notice is that the two eyes don't have identical images. They're slightly different, and we call this disparity. We also know that the right side of the visual field is now being processed by the left, and then vice versa. And as we move to the back of the visual cortex, things get really, really messy. Not only is right left and left right, but up is down and down is up. So it's flipped. So it's flipped left and right, and it's flipped up and down, right? Why? I have no idea. But we know that clinically this is very, very important. The last thing I'll mention that's quite interesting is you'll notice that the fovea part, the central part of the visual world, this sort of central 10 degrees, has a lot more real estate than the periphery does. Notice all this area here, this ring, has a lot less real estate in terms of the visual cortex than the center part of the visual field. So in other words, the brain dedicates more machinery, more brain power to that central visual area that you're looking at, and a lot less information to the periphery. All right, we'll get back to why this is an important point. So just a takeaway organization, takeaways: right is left, left is right, up is down, down is up, and the brain dedicates a lot more hardware, a lot more machinery to that central visual area compared to the rest of the visual field. Here's an important point to think about when it comes to this anatomy, the fact that there's this crossing over. The reason why that's clinically important is that if I were to look at a patient and they tell me they really don't see well in one eye, but they see just fine in the other, 99.99% of the time, that's a problem with the eye. I know that the brain is probably okay. In contrast, if one patient tells me, yeah, I don't see so well in this eye, I'm missing one side, and then when I close my other eye, I'm still missing that side, 99.99% of the time, I know it's the brain. And that's a very, very important distinction to think about from a neuroanatomical standpoint, because that's what the prediction suggests. If one eye has an impairment and the other eye doesn't, that means that that lesion is somewhere in front of the chiasm, right? Before things cross. If, in contrast, the patient describes the same type of visual deficit in both eyes, then I know that that damage was after the chiasm, after things crossed. And that's an extremely important anatomical landmark. And it's really interesting because a lot of patients with hemianopia, in other words, miss, once, let's say they're missing their right side, very, very often they come to me and they say, yeah, there's a problem with my right eye. I can't see on my right side, right? Then I ask them, close one eye. Do you still see your right side? No. Close the other eye. Do you still see your right side? No. I know it's the brain. It's not the eye. Right? Does that make sense? So it's a very, very important landmark to help you distinguish whether it's largely an eye-based problem or whether it's largely a visual brain or overall brain problem. So very, very important landmark to distinguish that. So again, just to reiterate, one eye okay, other eye not so good. That's probably in front of the chiasm. Both eyes showing similar deficits or similar impairments, probably a post-chiasm problem. Nice video here just to show you exactly what I mean. I think it illustrates how information leaves the eye, enters the thalamus, as I mentioned, and then projects to the back of the visual brain. You can see here, this is the eye. Sorry, let me point to you. The eye information leaving. There's the chiasm. You can see that split of information when it comes in to the back of the brain. So just a nice video showing that at the chiasm, things cross and they're maintained until they get to the back of the brain. So everything in front of the chiasm leads to one prediction. Everything past the chiasm leads to another prediction. So important landmark from a clinical standpoint. Very, very quickly, the overall anatomy of the eye. Again, I'm sure you spent a lot of time with this. Just very, very briefly, there's two ways to think about the eye. There is the optical component, which has to do with the cornea and the lens, how information or an image is focused onto the back of the eye. You know that some people are nearsighted, some people are farsighted. Farsighted individuals is called hyperopia. These are individuals where the image does not land to the back of the eye, it actually goes too far. So we use correction, a positive lens, to bring that image to where it's supposed to be. People who are nearsighted or myopic, that image focuses in front of the retina. So we use a minus lens to bring that image closer to the retina and focus properly. So that is the optical component of the eye, all right? The second piece of the eye is what we call the neural component. This is the light sensitive piece, and that's the retina, right? Again, just in the interest of time, I won't go into the anatomy, but you know that there are rods and cones. These are color and light sensitive cells that are distributed throughout the retina. We know that they're very, very dense in the macula where the images are typically focused. And we know that, again, this is the light capturing part of the retina of the eye that then sends that information to the back of the brain. Once the information leaves the eye, it enters the thalamus, as I mentioned in my first half hour, right? So information is focused to the back of the eye, passes through, focused onto the back of the retina, through the optic nerve into the thalamus, and then reaches the back of the brain, which is the primary visual cortex, right? And again, that's an overall organization that we see throughout many of the senses. So back of the eye, focus, optic nerve, entering the thalamus, and then the thalamus sending its projections to the back of the visual brain, the occipital visual cortex, right? During this period, these thalamic areas, and there are many of them, one's called the LGN, the pulvinar, superior colliculus, you have them here just for your notes. This is typically a relay station, if you will, of how information is entering the brain. So it is involved with things like contrast, it is involved with things like attention, it is involved with moving the eyes, that preliminary information that's entering the brain and being sent to the back of the brain for further processing, right? Once we're in the visual cortex, this is where the meat and potatoes really happens, right? This is where the processing really takes place. So we've now taken that information, we've entered the visual cortex, and remember, the cortex are where the executives live. Now we're going to really scrutinize and look at that visual information, scrutinize it, and make decisions. And the one thing that's really interesting about visual information is that once that image enters the visual cortex, the first thing the visual cortex does is break it down. It scrutinizes it in different aspects. We know that there's a part of the brain responsible for form, the overall shape of the object. We know that there's a part of the brain specifically responsible for motion, how that object is moving in space. We know that there's a different part of the brain responsible for the color of that particular object, right? So we know that once a visual image is focused onto the retina, is sent to the brain, the first thing the brain does, the cortex does, is break it down, right? So visual perception is not a passive process. It's about taking that information, scrutinizing it, and then putting it back together, right? So a little bit more information about that. Here are the visual areas that you may typically have heard in past courses and so on that give you an idea of how that information enters the brain. So back to our little guy, once it enters the primary visual cortex, that's kind of like a rough sketch pad of what the image looks like. Edges, some color, et cetera, et cetera. It's a very crude representation of what the eye has sent the rest of the brain. This is called V1, visual area 1. After that, we have what's called visual area 2 or V2. This is where the brain fuses the two images of the two eyes, right? This is where we get stereo perception and depth. So it's taking the two images, that crude image, and fusing them to give us this perception of overall depth. V3, we know, is responsible for form, the overall shape of the object. V4 is responsible for color of that object. V5 is responsible for the motion of that object. And we go on and on and on. We know, for example, one area called FFA. This is called face fusiform area. This is the part of the brain and temporal cortex that's only involved with processing faces. That's all it's interested in. Right? There's another part of the brain called EBA, which stands for extra body area, only responsible for biological motion and looking at how a biological movement happens, right? That's all it's interested in. These aren't relative to where they are in the brain, by the way. I'm just showing you how the visual brain breaks this all down, and they're all interconnected. So visual information enters the visual cortex, and the first thing it does is break it down and scrutinizes different aspects of the visual scene and then tries to put it back together, right? You'll see why that's important in just a couple of seconds. Just the last comment, I showed you about 6-7 different areas on the previous slide. We know today through neuroscience research that there's approximately 30 different cortical areas that contribute to visual perception, right? Lots of executives who are involved in this decision process. So again, entering the cortex, different areas, scrutinizing, looking at different aspects of the visual scene, and you can see that summarized for you as well. So again, it doesn't just enter the visual cortex exactly the same way that it entered the eye. It breaks it down and puts it back together again. And I'll give you some examples of why that's important, all right? Other thing to keep in mind, something that's probably relevant to what you know already, is we know that overall, the visual system at the cortical level is organized into a division of labor, right? We know that information that enters the visual cortex and goes up to the parietal cortex and ultimately into the frontal cortex, we call this the dorsal visual processing stream, or the WARE system. This is the part of the visual processing stream that's involved with figuring out where things are in space. In parallel, we have something called the ventral visual stream, or the what system. This is the repository of visual information in the brain. Faces that you've seen before, objects that you've seen before. This is the filing cabinet, if you will, of all those previous images. So the where and the what system are processed in parallel. The reason why we know this is because patients who tend to have damage to the parietal cortex can identify objects very, very easily, right? But they don't know where they are in space, right? They can't point to them in space, for example, but they can say, yeah, that's a key, that's a lock, that's no problem. In contrast, patients whose parietal cortex is intact but have damage to the temporal cortex, we call this agnosia. They can pick up the object very, very easily. They know where it is in space, but they say, I have no idea what this is, right? So that shows you that the two streams are handling two types of information. There is a caveat to this now, and more we're knowing is that these two streams talk to one another extensively. So while the where and the what pathway, or the dorsal and the ventral pathway, is useful conceptually, I want you to also be aware that it's not a hard and fast rule, and to say that these two streams do not talk to one another. They definitely do, right? This is an important concept because when we talk about CVI, what we suspect CVI is a condition that it predominantly affects the dorsal stream. And what we know in a lot of kids with CVI is they have problems with spatial processing. And we'll show you some evidence of why we think that's indeed the case. So useful to consider these two processing streams as parallel processing directions, if you will, but also understand that more and more we're starting to see evidence that the two talk to one another, okay? A couple of other things just to sort of wrap things up. This is a really nice schematic of the visual system simplified, right? To kind of give you an appreciation of just how complex all these visual areas are. What I'm showing you here in this little red circle, this is the retina to the thalamus to the primary visual cortex. In other words, the eye to the thalamus to the primary visual cortex. All this is the cortex handling the visual processing. So this information from the eye to the brain, that's just information coming in. To actually process that information requires all this above as well, right? So it gives you an appreciation of just how extensive the cortex is when it comes to seeing the world. So just understanding the eye entering the brain, entering the thalamus, entering the visual cortex, that's just a sliver. It's a very small proportion in terms of overall what the brain has to do in order to see the visual world or to process the visual world. Important thing to take away from that. A couple of things now that I think will be interesting from a visual perceptual standpoint. Many of you have probably seen this visual illusion. It's called My Wife and My Mother-in-Law. So some of you see a young girl turned away from you, and some of you see an old woman. Am I right? Everybody kind of see that? Ashley, I'm going to ask you to jump in on this one. It's a little bit hard to do by Zoom, but Ashley, who do you see?
I see the girl turning away.
You see the girl turning away. Interesting. Okay, I'll tell you why that's interesting in just a second. But I'm guessing that some of you see the young girl turned away and some of you see the old woman, correct? Hopefully that's true. This is a great example of what I mean by processing. I see a chat there. Do you want to jump in, Ashley, and add what somebody said?
One person saying I see both, but there is a question related to when you would see a neuro-ophthalmologist versus just an ophthalmologist, if you want to do this part and then come back to it.
Absolutely. Can we hold that question for the general question period? Good question, certainly a good question, but a little bit of departure of this conversation. That's okay. Sure. So Ashley saw the young girl, right? Some of you also saw both. Some of you also saw the older woman. It turns out that who you see is correlated to how old you are. If you're younger, you're more likely going to see the young girl. If you're older, and I'm not going to say what older means in this particular case, you're more likely to see the older woman. So there's a correlation with this. Why is this important? The reason is this image never changed. The image that's falling onto the retina and entering the visual brain is identical no matter who you are. But how you perceive it, how you interpret that visual image is a product of your brain handling all that information. And interestingly, it seems to be correlated to how old you are as well. So this is what I'm trying to say by visual processing. The way that you perceive the world is very much a reflection of the world you've already seen. right? So we greatly influence, we give our take to the information that's entering the visual brain. So just a nice example demonstrating how that's true. Again, remember, I didn't change the image. I'm not making it more likely to see one versus the other. You are deciding, your brain is deciding which one that it's seeing, right? Any questions, any comments? I saw a couple of flashes. I don't know, Ashley, if you want to check.
They're just helping me find the older lady. Sorry.
Someone is, oh, you want to see the old lady?
I
Very
couldn't
good.
find it.
Well, take that as a compliment, Ashley. How's that? Okay.
Sure, I'll take it.
So you saw, so this is the girl's cheek that's turning away from you, right?
Yes.
Yeah. Here's the old lady. That's her nose. Those are her eyes. That's her mouth, and that's the chin.
No, I guess I can see it. Yeah, I guess.
Yeah, you see it now?
Mm-hmm.
Okay.
Now I can, yeah.
Okay. I see both. I have no idea what that means. But the other thing that you'll notice is you cannot see both at the same time, right? Your brain has to commit. You see either the young girl or the old lady, but you never see both at the same time. Again, your brain has to decide and make a judgment, make a call based on the information that's entering. And visual illusions are essentially that. It's your brain forced to make a decision based on the information that's coming in. All right. Let's go a little bit further, I think, to kind of explain how this comes together when it comes to eye movements and processing information in general. The important thing to realize, and this may be kind of striking, is we do not notice or process the majority of visual information that enters the eyes. People find that hard to believe. They think that if their eyes are open and the information is coming in, they see everything. It's simply not true. And it's too bad because during the regular course, I have all these sort of blind sights and various demonstrations, but it's difficult to do, obviously, through the computer. The point here is that there are limits to the processing capacity of the visual system and brain. And let me give you a simple example, right? If you hold your hand out and put one M&M, you can count that very easily. 2, very easy. 3, 4, 5, very easy. By the time I get to 7 or 8, you start losing count, right? And you have to serially start counting how many are there. That's just a simple demonstration of the actual conceptual limit of the visual system. There's so much information it can handle at a glance. After that, it becomes a lot more of a brain power sort of issue. That's an important thing to think about. Also, when we talk about CVI in terms of complexity and clutter and things like that, kind of relates to that conversation about just how much can the brain handle at any given time. That being said, the other thing to remember is, as I showed you, the brain dedicates a lot more information to the center of where it's looking compared to the overall periphery. It doesn't have a high resolution picture of a visual scene. So for example, if I were to look at this picture here, that's not what your brain sees at any given time. What your brain really sees is only a small part at high resolution, fine detail, and in color, and the rest is kind of blurry, right, in the periphery, at a snapshot, a millisecond of time, if you will. The reason is in order to create a visual scene or a visual image, what the brain is doing, excuse me, is moves the eyes. So what the brain is doing is taking millions and millions of snapshots of a visual scene and then stitching them together so that it actually creates a representation of the visual world. And this anatomically makes sense. If the brain had to see the entire visual world at high resolution, your visual cortex would be this big, right? It would need a massive amount of brain power to be able to process the entire visual scene at high resolution. And that's just not efficient. So what it does is it dedicates brain power to the central part, has a gist of what's happening in the surrounding, and then it moves the eyes, as I show you in this slide here, right? So the solution to solve this problem is to move the eyes very, very quickly onto the visual scene. And then over time, the brain is stitching together a high resolution representation of that scene, right? The other thing that's interesting is that eye movement patterns will reflect the content and the saliency of the visual scene. In other words, the eye movements are not random. The reason the eyes move a certain way and in a certain direction reflects what your brain and state of mind is. And I'll show you more examples of that in just a second as well. So the eye movements are meaningful. They're not random, right? It's not like I'm just going to randomly look at the visual world and try to create. It actually has purpose. There's a method to the madness, so to speak, right? So in the end, in reality, the brain decides where to look and what we should pay attention to. And again, this goes back to this multi-level organization. Information is coming in, the brain is looking at it. That's interesting. I need more information. Move the eyes. That's going to go in. I'm going to update, and it continues and it continues and it continues and it continues. So it's this constant sampling of information that it does in order to move forward. Let me show you exactly what I mean by this eye movement issue. This was a traditional, or I should say a classical piece of work by a guy named Yarbus, who was tracking eye movements. And he showed this very, very simple picture to subjects where this gentleman walks into a room where people are perhaps having dinner, right? He then tracked the eye movements and said, I want you to tell me to just freely explore the visual scene. Nothing special, just look at the visual scene. And where do you notice the eye movements are? They tend to be on the faces of the individuals, right? Spending less time on the walls and chairs and things like that. The brain automatically goes to things that are interesting and meaningful in the visual scene. Then he said, I want you to tell me, what is the material wealth of the family in the scene? And what do you notice? They get off the faces and they start looking at what the people are wearing, right? Exactly the same visual scene. I have not changed the picture, but where the brain is deciding to look now is driven by the information that it wants, right? So again, nice example that the way you see the world is a product of the information that the brain is seeking, and it's a construction of the information that's coming in. So just simply changing the instructions to the person changes the way that the eyes move, right? He never said to the subjects, oh, I want you to look at their faces, or I want you to look at their, or what they're wearing. But notice how dramatic the eye movement patterns are simply by changing the question of what they should be looking for, right? Or the judgment they should make from that visual picture, right? Another thing you probably all know, right? Making eye contact. We know that during developmental periods, as a child gets older and older, we tend to be very, very good at maintaining eye contact, right? What do we know about children who are on the spectrum, who have autism spectrum disorder? They don't maintain eye contact. That's indeed what the eye tracking tells you. They'll look almost everywhere else except the eyes. So again, eye tracking is very, very useful as a window into the brain as to what they're looking at. Okay, and we'll get back to this in the case of CVI in the second half, right? Here's a little demonstration, and we'll wrap up the first half in just a couple of minutes. Again, it's going to be tough to do by Zoom, but I think this should work. Very, very simple task. I'm going to show you something, and I want you to find the blue O. All right, and actually, you're going to be my guinea pig. Okay, so when you find it, I want you just to raise your hand. Okay, ready? Here we go. All right, pretty easy, right? There it is. Okay, let's do another one. Find the blue O. You ready? Pretty easy, right? There it is, right? Here's the next one. You ready? Find the blue O. Tick, 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 tick. That's my point. That's my point. A lot harder, isn't it? In fact, just to prove I'm a nice guy, there's two of them, right? A lot harder, right? Why? Let's break this down based in terms of the integration theory. When I asked you to identify the blue O and everything else was the same except the shape, you were essentially using your shape area, your form area. Only one executive had to really make a decision on this, right? When I asked you to find the blue O and everything, the shapes were identical, it only was different in terms of color. Your color processing area was the executive and made the call now, right? When I ask you to now find the target and I've mixed the shapes and the colors, now that's two areas that have to talk to one another and get on the same page. And that's the delay, right? That's integrating. Now the executives have to talk to one another and make a decision. You saw how quickly that delay changed just for a very, very simple task like this. Imagine what the brain has to do to be able to perceive the real world. Thinking of in terms of color, in terms of shape, and things of motion, of depth, all the cues that make a very, very rich visual scene. The brain is constantly integrating that information and making a call, right? And it has to do it quickly because the visual world changes very, very quickly as well. That being said, what we can predict is that if the brain has an impairment in connectivity, if the brain isn't wired right, we should anticipate problems with visual perception, shouldn't we? Right? And that's exactly what CVI is. CVI is a condition where the visual system and the brain overall is not connected in an optimal way. And we think that is related to the visual problems that they have. So hopefully that demonstration convinces you the importance of integrating information. It's not a passive process. The brain has to do work. And the harder the task, the more it's going to have to work, obviously. But it's still really remarkable how quickly and effortless visual perception is. You just open your eyes and you see the world around you. Yeah. Okay, so that's the first section. I think we're still doing okay in terms of time. Any initial questions about this first section? I know, Ashley, you mentioned about neuro-ophthalmology versus just a regular ophthalmologist or optometrist.
Sure. Sherry Lowe is asking, with all the factors you've mentioned, physiology, development stages, disparity, when should someone be referred to a neuro-ophthalmologist versus just an ophthalmologist? My experience is usually only when you know that there has been a definite TBA, either from stroke or trauma.
Yeah, I would give a different perspective on this. And this happens also from my experience working with parents and teachers as well. In my personal opinion, it's not the specialty that matters. It is the familiarity of working with individuals with visual impairment, particularly when it comes to ocular versus brain-based visual impairment. There are plenty of optometrists, there are plenty of ophthalmologists who are not necessarily neuro specialties, who are very familiar with CVI, for example, and I would be more comfortable going with them because they are familiar with the condition. They realize that it takes time. They realize that time has to be spent. Whereas a neuro-ophthalmologist, in principle, certainly knows the neuro part, but if they're not familiar, for example, with CVI or brain-based visual impairments, how much more does that get you forward? And the reason why I say that is particularly, again, I'm jumping to the particular case of CVI, is I really believe that the providers have to be advocates. It's not just a question of assessment. They have to get behind the patient and the family to be able to support anything that they're looking for from a support standpoint, from a registration standpoint as well. So I don't see it necessarily as a specialty issue. I see it more as a familiarity issue. So the specialty and the letters after the name for me I don't think are as crucial as it is an individual who has familiarity with the condition and is willing to act as an advocate beyond just the exam itself. That being said, I do work very, very closely with the division of neuro-ophthalmology where in my hospital. And typically the neuro-ophthalmologist, that is correct. They think about the eye and they think about the brain and the whole system. That's absolutely true. But at the same time, if this neuro-ophthalmologist is not familiar with, say, something like CVI, I don't know how much more useful that necessarily is, simply because they've had that training of the eye and the brain. Does that make sense?
It does to me. The next question comes from Pam Arbeiter. Would someone with dyslexia have connectivity issues as well?
Yeah, great question. And a lot of evidence is suggesting that dyslexia seems to be related with impaired connectivity of the brain. What areas, et cetera, and so on, we don't know. But this is a general term when we think about developmental and brain disorders on the net. There is something called the Human Connectome Project. You're all familiar probably with the Human Genome Project, where we mapped out the chromosome areas of the human genome and know where different genes are on various chromosomes. There is something now called the Human Connectome Project. It's actually one of the biggest sites is in St. Louis, by the way, at WashU. And what we're doing there, I have nothing to do with it. What that university is doing there is mapping the connectivity of the brain and associating the wiring of the brain with various conditions, Alzheimer's, dyslexia, Parkinson's, et cetera, because the overall theme is that if the brain isn't working correctly, it's probably related to the fact that it's not wired correctly.
So the next question comes from Jessica Gibbons. Any thoughts about mental health trauma and its relevance to CVI? Think reciprocal blindness.
Yeah, absolutely. Great question. If it's okay, can we save that till the end after our presentation of CVI?
Absolutely.
Yeah, because I think that's a very, very good and relevant question and kind of gets back to, as I said, this emotional part of the brain as well. So yes, very, very relevant. But if it's okay, let's leave that for the end of the second half.
Great. So did you want to take a short break?
I'm completely fine. I can keep going. This is the first lecture I've ever given wearing my slippers, so I am super comfortable, ready to go. It's up to you guys.
We typically just keep on going, but I'm not the one doing all the talking and no one wants to hear me.
Oh, no, don't worry. I'm used to talking. Not a problem.
I'm getting some keep going from.
I'm getting keep going. All right. Okay.
Yeah, we're ready. We want more.
Again, either they like it or they just want to get this over with. Okay, that's fine. I'm open to both options. All right, so let's start the second half now. I want to concentrate more on this idea of neuroplasticity, and also specifically as it relates to visual impairment, blindness, and also CVI. And I will end on hopefully what I think is something useful for all of you in terms of what I call the laws of plasticity, or how I think we can leverage brain plasticity in terms of brain development as well. Not just in the case of CVI and blindness, but just overall development in general. Yeah. So neuroplasticity, very, very, lack of a better term, a sexy term. You hear this in companies now who want to sell you all sorts of programs. You hear this in apps. You hear this on TV programs and so on. And everybody gets excited when they hear this term. So what exactly does neuroplasticity mean? So neuroplasticity comes from the Greek term plastikos, which means to mold or to shape, right? And in the same way that a piece, it's where we get the word plastic from, by the way. And in the same way that a piece of plastic can be molded and shaped into something else and serve another function, so too can the brain. And that is exactly what we mean. Neuro referring to the brain, plasticity referring to the term plastic, meaning to change its shape and its function under the right conditions, right? So that's what neuroplasticity means. We'll get a little bit deeper into that. And here's a formal definition. Neuroplasticity is the ability of the brain to change its structural and functional organization. Notice I have the two terms underlined. It's structural and functional organization in response to development, experience, the environment, or damage, right? It's a general term of what neuroplasticity means. One thing also that's important to keep in mind is it is not a guaranteed fix. It is the inevitable consequence of how the brain works throughout a lifetime. Right? The reason why I say this is very often I'll be working with patients and their families, right, after a brain injury or something like this, and very often the family will reach out and say, so when's the neuroplasticity going to kick in? Like when is this all going to just get back to normal? As if neuroplasticity was some sort of magic bullet or magic fix. It's really not that way. The way to see neuroplasticity is it's an intrinsic property of the brain. Neuroplasticity is a fact of life, right? Death, taxes, and neuroplasticity is the way I see it, right? All of these issues come into play throughout a lifetime. The brain is constantly changing its structure and its function in order to change its brain wiring, its composition, the way that it interacts with itself in response to the outside world. Another way to kind of think about it is the mind or mental processes represents an internal model of the outside world. And it's how you interact with the outside world. You take information in, you build that model in your mind, and you act on it. You make predictions about that model. You reach out for something, you wait for something, you run away, you run towards it, you run away from it. It's all about creating an internal representation of what that outside world is to you. And neuroplasticity is the mechanism that allows that to happen. If you really want to stress the system, a brain injury, sensory deprivation, for example, that is really where you see neuroplasticity at its best, so to speak, right? It's really extreme pressure, if you will, for the brain to change its overall structure and function. So it's important to realize neuroplasticity isn't necessarily a good or a bad thing. It's a fact of life, right? Let me give you an example of what I consider my favorite example of neuroplasticity. If you ever come into my office, you'll see this picture hanging on the wall. I've had this picture for many, many years. It was taken by a photographer named David Seymour. It was taken after the Second World War in Europe. And what this photographer did is he went around trying to take pictures capturing the plight of children after the Second World War, after all the bombings and destruction, and how these kids were kind of handling that really tough situation after the Second World War. He came across this young boy who was blind, right? And this was in Italy. And like many children of his age who were born blind, he learned to read Braille, right, as a form of communication and language. One day that child was out with his friends. He was playing in a field in a place that he should have been playing, and he picked up a landmine, and the landmine exploded. It destroyed both his hands, and he became a double amputee. He still reads Braille. That's what you see in this picture. Not because he sees the dots, but he's learned to read Braille using his lips and the tip of his nose, right? This is another example to kind of show you what happened. Here you can see he's a double amputee. They've enlarged the Braille print for him, and he uses his lips and his nose to read the Braille print, right? This is neuroplasticity. This is exactly what I mean. A tremendous stress, a tremendous challenge to the brain. The brain is now trying to figure out a solution, is trying to adapt to a challenge by changing its structure and its function to respond to it. That's really what I mean by neuroplasticity. So really a dramatic example. Let's go a little bit more into the situation of ocular blindness and ocular visual impairment, something you're all very, very familiar with. Here's an interesting sort of philosophical question. Stevie Wonder, who you know, was born blind, or not congenital. If my memory serves me well, he had glaucoma, and I think he was visually impaired at birth, but really became profoundly blind in his early years and teens. And as you know, became an extremely successful musician and wrote many hit records and things like that. The question I have for you is this: if Stevie Wonder didn't lose his sight, or didn't essentially lose his vision at a very young age, would he still be an extremely talented musician that he is today? So in other words, did blindness somehow confer an advantage, right, to his overall development? I think it's an interesting philosophical question, but we know that we can try to answer this from a neuroscience standpoint as well. We know that there's a lot of evidence of enhanced compensatory behaviors in individuals who are profoundly blind due to some sort of ocular damage or ocular pathology. We know, for example, there is evidence of people who are born blind that have a keener sense of touch, a keener sense of localizing sound in space, are able to identify smells in space as well, and other senses as well, not necessarily just I should say sensory modalities, but also have enhanced verbal memory and recall. For example, you can ask a blind individual to recall 10 words, come back to them a month later and ask them to recall those 10 words, and typically they do better than aged match-sighted controls, right, on this task. So there seems to be evidence of these compensatory behaviors in individuals who are born profoundly blind due to ocular cause. There are caveats to this observation, however, that are important to realize. First of all, this isn't in every blind individual, and for reasons we don't know why, right? This seems to be in certain individuals, but not others, and we don't have an understanding of why this isn't universal. The second thing is that more often than not, these enhanced behaviors are observed under very specific and controlled experimental conditions, right? It doesn't mean necessarily that in the real world, these advantages are really obvious, but under controlled conditions, we know that this seems to be true. And the last thing I'm going to mention is this really enhanced behavior in the blind, or is this sighted people who are just really bad when you blindfold them? Right? So there's other side of the coin that you can actually interpret this by, right? Perhaps ocular blind individuals are operating at a certain level, but when we ask a sighted person not to use their sight and use this task, that somehow puts them at a disadvantage. So just important thing to think about. There is evidence of this compensatory behaviors, but it's not a hard and fast rule, and there's still things we need to figure out about this. Another question I think that's really worth answering is that in this particular situation, if you have an individual who's born profoundly blind because, again, damage or disease to the eye, what is the fate of the visual cortex, right? You remember in the first hour, I showed you that the visual brain is about 25, 30, 35% of the overall cortical surface. That's a lot of real estate, right? What is the fate of the visual cortex, or the part of the brain that is, quote, supposed to be involved with visual processing, in an individual who has no sight, right? Does it do something else? Does it just waste away? Does it stay silent? Well, it actually does something, because one thing we do know about Mother Nature is she doesn't waste brain cells, right? So this is how we've answered it. And we've used a technique called functional magnetic resonance imaging, or functional neuroimaging in general. You remember that psychopath example I gave you in the first hour? It allows us to find these areas of hotspots of activity, if you will, highlighting brain activity areas in response to a particular task. So let's look at this example. Here you see what's called a sagittal projection. So in other words, I've taken a cut this way through the brain. This is the occipital cortex here. This is theoretically where the eye would be in this individual, right? And we see this robust hotspot of activation in the visual cortex. These individuals who were in the study were all born congenitally blind, born blind with profound ocular blindness. So the question is, why would the visual cortex be active in a person who's never had sight? And the answer is this: these individuals are reading Braille, right? So we know that in the case of profound ocular blindness, if you are a proficient Braille reader, the visual cortex now becomes responsive to tactile stimulation, particularly if it's related to language. How do I know this? You see this activation that I see here? This is the motor cortex. That's actually the representation of the index finger, what they're using to read the text. This little other hotspot of activation, this is parietal cortex. That's the part of the brain responsible for spatial processing. So very, very intriguing that in the case of ocular blindness, congenital ocular blindness, the visual cortex becomes something else. And it's not just Braille reading and touch. We know that this is true for sound localization. We know it's true for smell. We also know it's true for non-sensory things like verbal memory, recall, and language as well. So it's an important takeaway that the occipital visual cortex is the seat, or the site, I should say, of compensatory non-visual behaviors. So the visual cortex, in the case of ocular blindness, becomes recruited to process non-visual information. Like I said, Mother Nature doesn't waste brain cells. She uses that information and uses that area to process non-visual information. And we think that this activity may be the marker of why some individuals do better and others don't. So it's kind of it could be an index, if you will, of these compensatory behaviors that we see. Because in some individuals, there's actually a linear relationship. The better you're reading Braille, the more activation, for example, we see in this area. Again, not a hard and fast rule, but it's an interesting index of what's happening there. Here's an interesting case study. I had the pleasure of working with this patient in my first or second year of fellowship, just to answer the question, well, how do I know you're not just, this guy coming from Boston with his fancy brain pictures and Photoshop, and how do we know this is actually true? How do we know that this activation is actually useful? So let me show you a case study where I hope actually drives this point. So this is a case report. This is something that happened about 20 years ago. I'm telling you the story the way that I heard it. I wasn't there when this happened. I did work with this patient, but I'm telling you the story as it occurred or how it was told to me. So at the time, this was a 63-year-old right-handed female. She was born, she was blind at birth. She had retinopathy of prematurity, as you know, that has, that compromises her overall visual acuity. Her reported visual acuity was no light perception in both eyes, so profound congenital ocular blindness. She hits normal milestones, and she learns to read Braille at the age of 6. She's a very, very proficient reader. I think you'll agree with me, those of you who are familiar with Braille reading, 120, 150 symbols per minute is a very, very fast Braille reading speed. She was a highly proficient Braille reader. In fact, she was the editor for a Braille journal in her native country of Spain. So she was reading Braille every day. She's very, very familiar with Braille. One day she wakes up. She doesn't feel very, very good. She's complaining of lightheadedness, difficulty swallowing, and then she loses consciousness. She goes to work nonetheless. And when she gets to work, she loses consciousness. They rush her to the hospital, to the ER. She falls into a coma for 24 hours, right? And then she wakes up, she's alert and interactive, and she's given, quote, a normal physiological and neurological exam. She's fine. She just woke up into the hospital. The interesting thing is this: she asked for her phone book, which of course is written in Braille, so she can contact family members and explain to them what had happened, that she's in the hospital. She takes the phone book, she starts reading the dots, and she says, you know what? This is really, really strange. I can't understand anything that's written here. I feel the dots, it's definitely Braille, but I can't understand anything that's written. What happened? Turns out she had a stroke. Anybody want to guess where that stroke was? In her visual cortex, right? Not the part of the brain responsible for language, not the part of the brain responsible for memory, not the part of the brain responsible for touch. Theoretically, the part of the brain that she wasn't using anymore, her visual brain. So this is an axial MRI scan. This is what's called a T2 weighted scan. In white, you see dead tissue. This is infarcted tissue. These are just 2 cuts here, right? To this day, this woman is alexic, meaning the acquired inability to read Braille, following stroke to her occipital visual cortex, right? She lost the ability to read Braille because of damage to her bilateral damage to her visual cortex. So a tragic story, but certainly very, very compelling in terms of telling us that this recruitment, the fact that the visual brain is being used by other senses, is actually important, right? It's not an artifact. It really is the brain shifting and changing its rewiring, its connectivity to process that information. And to this day, she is unable to read Braille. Okay, so pretty dramatic example that indeed the visual cortex is important, right? So is all plasticity good plasticity? I think it's important to have this point. As I mentioned, plasticity is a fact of life. And just like I can show you these dramatic examples of compensatory or quote good plasticity, there are also examples of maladaptive plasticity as well. And a great example of that is phantom limb pain. If you're not familiar with this situation, typically what happens is an individual will have a limb that's amputated, maybe the hand, maybe below the elbow, or perhaps a leg. And what's interesting in these patients is despite the limb being missing, they will complain of extreme pain. They will say, I feel like my hand is being crushed in a vice, even though the hand is no longer there. So it's the brain trying to compensate for the fact that no information is coming in anymore, right? So that's a great example of maladaptive plasticity. It's the brain trying to figure out, this limb is missing. The easiest way for me to understand what's going on is to signal pain, for example, as an alert to the person. And it's very, very difficult to manage. These patients who have phantom limb pain are very, very difficult to manage because it's quite dramatic. It's quite debilitating, as if already they have to learn to use an artificial limb or something like that, and they're also dealing with chronic pain. It's very, very difficult as well. So a good example of maladaptive plasticity. It can go the other way. So let's get into now the situation of CVI. So I hope I've convinced you with a couple of slides that in the case of profound ocular blindness, the visual cortex becomes something else. It becomes responsible for processing non-visual information, touch, hearing, smell, things also like memory and language as well. What about if you had an individual whose eyes were normal at birth, but instead had damage to their visual cortex? Now, what can you say in terms of plasticity? How does the brain adapt and rewire itself when the hit is not at the level of the eye, but the hit is rather at the level of the visual cortex? And that is essentially what CVI is, right? So now we'll talk a little bit more about this particular situation. Let me give you a little bit of history and background in terms of how I got involved with this particular population. So some of you may know this institution. This is the Perkins School for the Blind. It is the first and oldest school for the blind in the United States. It's located in Watertown, a little bit outside of Boston. As I mentioned, this is where I did my internship when I was in optometry school. And again, certainly had a massive influence in terms of influencing my clinical career as well as scientific career as well. If you were to look at the classic Perkins kid, say 30, 40, 50 years ago, you would find children who were enrolled at the school who had ocular forms of blindness. In the early years, a lot of them had infection, things like syphilis, for example. Rubella was another common, so infectious causes that created blindness. Later on, that changed a little bit, things like retinopathy of prematurity, various genetic diseases like Leber's congenital amaurosis was quite common. And then what we noticed about, say, 20 years ago, that profile changed. And that profile changed in the sense that a lot of the children who had ocular blindness were now being mainstreamed, were being sent out in district schools. And the classic profile of the Perkins kid was not visually impaired necessarily to an eye problem, but was visually impaired because of a brain-based problem. Now, this was absolutely crucial, and I know I'm preaching to the choir, but this was a big, big issue because what the teachers noticed is all the things that were tried and true. For example, using a cane, reading Braille, orientation and mobility. The kids with these brain-based visual problems seemed to struggle with it. They weren't really responding very, very well to these classic curriculum strategies, even in the case when kids had quote usable vision, say 20/40, 20/60 visual acuity. So they had quote good usable residual vision function, but yet we're still struggling with basic skills like orientation and mobility, learning Braille, et cetera, et cetera. So why is that? How is it that you can have two kids, two with the same visual acuity level, one eye-based, the other brain-based, excuse me, and yet they're so different in terms of their education and their response to rehabilitation or rehabilitative programs. And that's when Perkins reached out to me and said, you know what, we need to understand this from a brain-based level. And that's very much how the project got started. A couple of facts that you might find quite striking if you don't know this already. This was a study done by Kong. This was in the Journal of APOS in 2012. They looked at 3,000 children who were enrolled or schools of the blind throughout the United States, and they rank ordered the diagnoses of the children who were enrolled at the various schools. And what did you notice? CVI, cortical or cerebral visual impairment, was the #1 diagnosis across the United States. The majority of the kids, no, excuse me, not the majority, the most common diagnosis, I should say, of the children who were enrolled for schools of the blind across the United States had CVI. I can tell you from my numbers that I know at Perkins, it's ballpark about 45, close to 50% of the children on campus. Their primary visual impairment diagnosis is CVI. What else do you notice here on this list, right? Optic nerve hypoplasia, retinopathy of prematurity, ROP. Turns out we did that, right? We were giving too much oxygen to these babies, and that's why the retinas didn't develop. Fortunately, we've learned from our mistake, and now we know that ROP levels are going way down. That's a good thing. Other things we noticed, things like syphilis, rubella, cytomegalovirus, toxoplasmosis, these are all infectious causes of blindness. Again, with good immunization programs, good public health programs, again, we should see these numbers to continue to decrease. Things like retinitis pigmentosa, Leber's, for example, these are congenital retinal dystrophies. In other words, genetic problems at the level of the retina. And again, probably many of you know that there's a lot of exciting research being done in gene therapy that could potentially reverse or stop or halt, I should say, the progression of these retinal diseases. The point I'm trying to make is as time goes on, there's going to be a huge rift between CVI and the rest of these ocular causes. We're going to get very, very good, and we're going to get these numbers down really, really low. But CVI is basically going to be on its own. And that's something really, really important to think about. It's the #1 cause of pediatric visual impairment in developed countries, including the United States. Not developing, developed. And the reason why we think this is so common is because most of these children are either born premature or born with some sort of perinatal complication. And the reality is we're getting very good at saving them. These kids were not surviving 30, 40 years ago, but they're surviving today in big numbers. They're thriving. And the question is, what are we going to do for them, right? We have to change the way that we assess them in the clinic. We have to be able to change the way that we think about what our expectations are in terms of these children. We have to change the way we teach them. We have to change the way we rehabilitate them. And we have to get on board as a group from different specialties to create a whole new strategy, a new pivot as we move forward in terms of dealing with congenital visual impairment, something very, very different than what we've done in the past. And in my mind, that's a really important situation to think about. Again, just to drive home this point, the #1 individual cause of pediatric blindness and visual impairment is not due to disease of the eye, but rather damage to the brain. That's a pretty profound statement. Very recently, the NIH, which is the National Institutes of Health, National Eye Institute specifically, wrote a very, very large opinion piece specifically on this. There are children, for example, who have very, very good visual acuity, 20/20, but yet clearly have all sorts of visual impairments. What do you do for these children? Theoretically, they don't qualify for benefits if their acuity is, quote, too good or don't have large visual field deficits, but they're clearly visually impaired. And we've heard horror stories, literally horror stories of families taking their children to eye doctor, to eye doctor, to eye doctor, to eye doctor, who looked at the eye, everything looked good. They read the letters on the chart, and the doctor said, I think your child has psychiatric issues. Maybe your child has autism. Maybe your child has behavioral issues. And the child went on a completely different trajectory simply because that eye doctor or clinician or doctor just simply could not realize that this wasn't about the eye, it was about the brain. So back to that earlier question, should they see a neuro-ophthalmologist? I would say they should first see a specialist or an eye care provider who understands what CVI is to begin with. All right, a little bit more information about CVI. I apologize if this is a review for most of you, but just again, so we're all on the same page. Cerebral, also called cortical, some of you might be familiar with that term as well. Also brain-based visual impairment. Cerebral is now becoming more and more the accepted term, particularly in Europe, in Canada, and the rest of the world. Cortical is still lingering, so to speak, in the literature in the United States, but there seems to be more and more this trend to use the term cerebral, largely because we know that cerebral visual, or CVI, is an impairment at the brain at all levels. Questions at the level of the eye, questions of the thalamus, questions of white matter connections, questions of the cortex. So it implicates the entire brain, and cerebral kind of encapsulates or captures that whole brain impairment, so to speak. And I think that's why you're hearing more and more the term cerebral being used. The old definition was abnormal visual function despite, quotes, a normal eye examination, to distinguish that what this child was going through, the visual impairment that this child was observing or going through, was brain-based, not eye-based. That certainly is true, but I don't think it's fair to say that the eye is always normal. We know that a lot of kids with CVI can have eye issues. They could have an eye churn. They could have reduced visual acuity. They could have, for example, retinal issues. But what we do know, or what the key distinction is those eye problems cannot be explained by a problem at the level of the eye only. They are clearly a brain-based problem. And this is a recent definition by Saki et al. In 2017 that I think is quite good. It says significant and verifiable visual dysfunction associated with damage to retrochiasmal pathways. Remember that magic word. The chiasm is either related issues versus brain-based related issues. We talked about that in the first hour. Retro meaning behind the chiasm. Retrochiasmic pathways and cerebral structures that cannot be attributed to disorders of the anterior visual pathway, meaning the eye and the optic nerve, or potentially co-occurring ocular pathology. That's not to say that an eye problem won't be there. It just cannot explain all the problems that the child is having, right? So a very, very key definition. Visual acuity can range from normal to profound blindness, which makes this very, very challenging, obviously. We see the whole gamut in the case of CVI. Often there are visual field deficits, and that's typically in the inferior visual field, and there's a reason for that, which I'll get to. Often we see these kids have reduced contrast, right? Often they'll tell us that they have a hard time going down steps, for example. They cannot see if things are not clearly distinguishable. Many will have ocular motorness issues, difficulty moving their eyes and maintaining attention, for example. The thing that is key when it comes to making the diagnosis of CVI, and this is a big challenge because there isn't a hard and fast or accepted rules or standards for making the diagnosis, is we know that a medical history has to be consistent with some sort of pre or perinatal neurological impairment or injury, right? Something had to have happened around the time of development, birth, or shortly thereafter, right? And we'll talk about what sort of things can happen. They typically will present with some sort of neurological impairment. It could be, for example, cerebral palsy in some cases, could be epilepsy, some sort of neurological manifestation, and characteristic what are called neuroradiological findings. Not all, but if we look at, for example, the MRIs of these kids, very often we'll see structural damage. But this also isn't a hard and fast rule. We see many kids who have clearly visual impairment issues, but the MRIs look normal. So that's not a hard and fast rule either to think about, okay? So it really is a clinical picture. It's a big puzzle that we need to put everything into consideration. Visual function, the history, understanding what type of deficits the child has, and trying to put it all together in a way that raises that clinical suspicion that this is CVI, right? The other thing that I'll mention that is really quite unique, and this is what our research really focuses on, is often there is the presence of what we call higher order visual processing deficits, right? Visual, spatial, and complex motion processing, as well as visual search and attention deficits. And this has been called dorsal stream dysfunction because they tend to be, we think, related to the dorsal pathway. Spatial issues, where things are in space and where the person, for example, is in space as well. This seems to be the predominant feature that we see. That's not to say it's the only one, but on balance, we seem to see these overall dorsal-like behaviors or impairments, I should say. So what does this mean? I'll give you some examples. Some kids, for example, tell us that they cannot walk and see at the same time. They'll be walking down a street and they'll walk by their home, for example, or they'll walk by their school or their store. They cannot sort of integrate all these motion signals that are happening in the periphery and then use that to be able to move their eyes and try to find a particular target in space. So they're very, very sensitive to motion cues, for example. A younger child might tell us that, yeah, you give them their toy and they say, yeah, that's my favorite toy. But if you put it in a toy box full of other toys, they can't find it, right? That's not a visual acuity problem. They identified the toy in isolation, but if you put it in a toy box with other toys, they can't find it anymore. We call them dumpers, right? They take their toy box, they dump all the toys out, and they have to spread them all out in order to find their favorite toy. That's not an acuity problem. That's a brain problem. That's a processing problem, right? Our older sort of teenagers will tell us in high school, walking down corridors during, when classes change over, they'll walk by a friend, they'll walk by their favorite teacher, they'll walk by their locker. They don't even pick up on the individuals around them. I have some kids tell me, it goes, yeah, my biggest issue isn't so much my sight, it's the fact that my friends hate me. They think I'm being being snobbish and I don't talk to them as I'm walking by. I simply just didn't see them. I didn't recognize them. Right. So again, higher order nature. This is not an eye problem. This is not a glasses problem per se. This is a brain processing problem. Barry Cran is the head of the Perkins Eye Visual Clinic at the Perkins School for the Blind. And he has a great analogy. He calls it the slot machine analogy because there is tremendous variability in these children. Every kid is essentially their own case study. It's not a universal thing by any means. So what he says is, imagine it's like a slot machine where each one of these tumblers could be an assessment. This one could be visual acuity. This one is visual perimetry. This one is higher, eye movements. This is contrast. And you pull the lever and whatever comes up is the combination for that child, right? And that's what really makes CVI so challenging, is there is tremendous variability in presentation. And there isn't just one fit and one fix for all these children. You really have to look and assess as comprehensively as possible each child, understand their strengths and quote their weaknesses, right? All right, a little bit of details in terms of what causes CVI. There are many, many causes, right? From a stroke, for example, that occurs while they're developing or shortly after birth. It could be trauma. It could be, for example, an infection like encephalitis or meningitis. We also know there are many kids who have CVI due to genetic or metabolic disorders as well. So this also makes it very, very challenging because the cause of CVI isn't universal or uniform as well. Many things can cause it. The most common is this, some sort of perinatal hypoxic or ischemic event. Essentially, the baby has a stroke during development. Like I said, shortly before birth or shortly thereafter. And what we know is the damage to the brain is extensive. It encompasses lower structures of the brain like the thalamus. It encompasses also white matter connectivity, and it also encompasses the cortex. Remember the building analogy that I gave you the last hour? This is very, very important to think about. All those pieces seem to be affected in the case of CVI. Just to break down this perinatal, I should say, hypoxia ischemic event, it's useful to break this down into two categories based on whether or not they're born premature or term. If they're born premature, in other words, before 32 weeks, this is typically associated with what's referred to as periventricular leukomalacia, or PVL. What happens is, again, the baby has a stroke and blood rushes into the ventricles of the brain. Remember that part of the brain I showed you that was fluid? It kind of helps the brain to absorb shock and so on. Blood enters that area because around the ventricles are very, very sensitive to changes in oxygen. So any slight change makes these areas bleed very, very easily. And as the baby develops, that blood is resorbed back by the brain and the rest of the body, and the ventricles grow larger. So you see these very, very large ventricles that appear in these children. I'm going to show you some MRI images of what that looks like. In contrast, children who are born term, we call this hypoxic ischemic encephalopathy, right? So they're born term and then have the stroke. Remember timing, very, very important. The damage is still very, very extensive, but doesn't seem to be as extensive as in the case of children who are born premature. We know that this typically affects areas of the brain that are called watershed. In other words, in between vessels, parts of the brain that are kind of too far to get their oxygen and their blood flow. So slight nuances that happen based on timing. As I showed you in the last hour, timing of injury is very, very important. So a useful way to kind of separate that out. So let's get now, like I said, back to the meat and potatoes. Let's look at some data and trying to think about how this all plays out. What I'm showing you here are 3 cuts of the brain. These are axial projections. So in other words, slicing through the brain this way. So you're, imagine slicing the brain like that. This is an individual with neurotypical development, has 20/20 vision. This is an individual with ocular blindness, has light perception in terms of their visual acuity. And this is an individual with CVI with 20/20 visual acuity. And notice these large ventricles that I mentioned to you, right? You see, these are the ventricles here, normally how they appear. Look how they look like in this particular individual. So classic example of PVL, right? What do you take away from this that's kind of striking, right? The person who has, quote, the most normal looking brain has the worst visual acuity. And the person who has, quote, the most abnormal looking brain has normal visual acuity. You see that mismatch? That's what makes this very, very challenging. And we call this in neurology, we call this the neuroradiological paradox, which means that lesion load, the extent of apparent damage that we see on an MRI, doesn't always correlate with clinical symptoms. And that makes it very, very challenging. In other words, even if the child is scanned by MRI and we look at it, it doesn't necessarily tell us what's going to be wrong with that child. We can get hints based on what we know about the structure and function relationship, but it doesn't really speak to these higher order issues and perceptual problems. So we need another way to look at the brain. And what I propose to you guys is to look at not just the overall structure, but the connectivity, the wiring. That should tell us maybe something. And this is what I propose to you guys. CVI is a disorder of brain connectivity, the way that the brain is wired. And this underlying connectivity should be associated with these clinical manifestations, these problems that these kids have. So what we need to do is not look at the overall general structure of the brain, the way that MRI does. We need to look at the wiring of the brain. So how do we do that? We use a technique called HARDI, which stands for High Angular Resolution Diffusion Imaging. This is a technique, to give you again very, very quickly the background. Instead of looking at the general structure of the brain, the way an MRI does, in this particular case, what the scanner is doing is tracking the movement of water molecules, right? We know that if a water molecule is moving randomly in 3 dimensions, it's probably in the ventricle. It's probably in a place that's not actual brain tissue. But if it's moving in a particular direction, back and forth, what we call anisotropic motion, meaning it's constrained somehow, we think that that water molecule is associated with an axon, with a brain cell. And what the computer is doing is taking that information, tracking that water movement, at millimeter precision, and connecting them through some very, very fancy computer work to give us a 3-dimensional representation of those pathways. And let me show you what that looks like. On the left, I have a control individual. On the right, I have an individual with CVI, PVL. This is an individual born prematurely. And I'm going to rotate these brains in 3 dimensions to show you exactly what the reconstruction looks like. So here it goes. So again, 3 dimensions, head to head, they're about the same age. This is the gray matter. This is now what the diffusion imaging looks like. These are the white matter connections. Then what we do is we ask the software, I want you to virtually dissect the rest of the brain and just show us what the visual brain is connected to. So here we say, look at the visual cortex, which is in the back of the brain here. What is it connected to? And what should show up to you? This pathway here from the back of the visual cortex to parietal cortex to the front of the brain is the dorsal visual pathway. It's called the SLF, right? Stands for superior longitudinal fasciculus. This is the neuroanatomical correlate of the dorsal or WARE pathway. This one, in contrast, from the occipital cortex down the temporal cortex is the ventral pathway or the what pathway, right? So we now have a way to disentangle the what and WARE pathway in a way that we could never do with standard MRI. And what does it look like in a CVI subject? The first thing you should notice right away is the ventral pathway is pretty good overall, but the dorsal pathway is dramatically impaired in terms of its development, right? Should be pretty obvious to you that the pathways are not affected the same way. So going back to this suspicion of dorsal spatial impairment or dorsal dysfunction seems to fit with this suspicion that the dorsal visual pathway is impaired in terms of its development, right? So putting some more numbers to that a little bit. Here is a sighted control, 17-year-old with 20/20 vision and neurotypical development. Again, the SLF is the dorsal pathway. The ILF is the ventral pathway. What do you think it looks like an individual who's born with ocular blindness? Pretty much the same, right? I challenge you to distinguish who is the person who's ocular blind and who's the person with neurotypical development, right? Both pathways look pretty good, right? Here it is an individual with CVI. Like I showed you, ventral pathway certainly not as developed, but the dorsal pathway is really, really impaired, right? This is an individual born premature. Here's another individual with CVI, exactly the same thing, but this person is born term. So it's not just a question of these large ventricles with PVL. Even in an individual who's born term, we see the same sort of selective disruption. The ventral stream seems to be largely intact, but the dorsal stream seems to be really, really impaired. This fits with the clinical presentation. If you talk to these kids, what will they tell you? I get lost all the time. When I'm in the car, I have no idea where the exit is, right? I don't like walking in busy crowds or busy places. Spatial related issues, right? So the anatomy seems to fit with the clinical presentation of CVI. Again, something you wouldn't see with a standard MRI. We can go even one step further, and this is called whole brain analysis or connectivity analysis, where we look at the entire brain overall, how it's put together. And this is what we do. You're looking again at the top of the brain. This is an axial projection. These are our sighted controls. This is about 15, 20 individuals who are age matched. And the blue dots that you see here represent different points of the brain. And the lines in between tell us how strong those connections are. Red means very, very strong. Green, a little bit, weaker. Blue, relatively weak. And in the sighted controls, overall, the brain is tremendously well connected. The overall connectivity of the brain is throughout and very, very uniform, right? How does it look in ocular blind individuals? Very, very similar, right? It's all there. In fact, if you mathematically subtract this brain from that brain, you actually see extra connections from the occipital cortex, the visual cortex, to language areas, to memory areas. So we think that in the case of ocular blindness, these extra connections could be the basis of these compensatory behaviors that we see. Wiring between the visual brain, auditory areas, memory areas, language areas, and so on. How does it look like in CVI? Very underconnected. Right? Lots of blue and green, not so much red, even though a lot of these kids have residual visual acuity of 20/50, 20/60, for example. So high visual acuity doesn't get you out of this, right? So it's an overall developmental issue. So back to my first question, how do you have two kids with the same visual acuity, one from ocular impairment, the other from brain-based impairment? Why are they so different? Their brains are connected differently. They're wired fundamentally differently. It's not an eye issue, it's a brain issue and how that information is put together. So hopefully that gives you some sort of compelling evidence as to why these two populations are fundamentally different, even though they may present, well, like I said, with the same level of impaired visual acuity. Okay, I'm going to stop there for a second before I continue. Ashley, any comments?
People are just hoping that it's being recorded because they want to watch again.
Okay.
And they're saying yes, that we're in the presence of royalty and we're not bored.
Okay.
I don't think there's any new questions, so
okay,
carry on.
Good, excellent. We'll continue on. Let me just wrap up in terms of the anatomy, and then I'm going to get to some functional and virtual reality work, which I think is going to be hopefully interesting for you guys when we kind of put it all together. Just in the interests of time, I want to show you that these changes at the level of CVR are extensive. They're not just at the level of the white matter. We know that, for example, the thalamus is reduced in volume in CVI. We also know that kids who are born premature have even more reduced volume than children who are born term. So what that tells us is that premature babies typically will show more impairment than term babies, pound for pound, even for the same nature or type of damage, because the thalamus seems to be more impaired in the case of premature babies. So at the level of the thalamus, we certainly see damage. We also see decreased gray matter. In other words, the thickness and the volume of the visual cortex seems to be impaired, not just in occipital cortex, but also in parietal cortex and temporal cortex as well. This also seems to be uniform in CVI. So there are cortical changes as well. And lastly, as I showed you, there are white matter connectivity changes. So back to our building, the lobby is impaired, the executives are impaired, and the elevators are impaired as well, right? So it's all throughout the brain that we're seeing these fundamental changes in terms of brain structure and anatomy, right? In the case of CVI. Okay, that was the anatomy part. And the last few minutes that are, or the last, I guess hopefully about 20, are we doing okay, Ashley? Yeah?
Typically, we go to 6:30, but
6:30. Oh, sorry, 6:30. Yeah, excuse me. That's right. I'm 7:30. I'm sorry.
Oh yeah.
Yeah, sorry.
I'm fine to keep listening. Whatever people want to do. There are a couple more things popping up now. People have some more questions. I think since you asked if people had questions.
Very good. I'm completely fine to continue. I've got about 5 or 6 more slides showing you some virtual reality and functional work, and then wrap up in terms of plasticity. And I'm happy to stay as long as you guys want to answer your questions, if you guys are okay with that.
I'm good with it. And then we'll just let it record. And if people have to go, they have to go.
Joyce, Jen, I see thumbs up. All right, keep going. All right, sounds good. Let's move now on to the functional assessment and what I mean by functional vision and visual function. I know they sound like similar terms, but they're very, very different, because really what we want to know is how a person sees, that's visual function, and how they use their vision in the real world, that's functional vision. And they don't necessarily go together, and that's extremely important. If you work with kids like CVI, like I do, you're going to hear things like this: dots, stars, and letters are boring. They do not want to sit and do standard eye tests all the time, like look at letters and the perimetry eye test and things like that. And even if you use a little bit more creative, so to speak, psychophysical stimuli, they just get bored. They really don't like looking at this stuff. It's just not interesting to them. Why? Because that's not what their visual world looks like. Their visual world looks more like this. This is what they're interested in, right? This is what they want to understand. This is what parents want to understand. But here's the problem. In the case of these well-controlled stimuli, you have excellent control in terms of the stimulus. You know the size of every dot, how big they are, every star, where they are, the luminance, the contrast, all the details of the visual stimulus you have very, very good control over. The problem is it doesn't reflect the real world. We call that ecological validity. It tends to have poor ecological validity. The real world, by definition, is ecologically valid, but you don't have control. There's lots of things going on here. It could be the lighting, it could be the overlap of the subjects, it could be, for example, the noise in the background. There's lots of things that could influence that child's behavior. So you have very, very poor control of the actual stimulus and task. That's the conundrum. What I propose to you guys is the middle ground of functional vision and visual function is virtual reality. We think that we can create environments that are more appropriate and at least mirror more the real world, but at the same time maintain good experimental control so we can tease these factors out. The second thing I'm going to mention that we study is eye movements. And you remember in the first hour, I talked a lot about eye movements and why they're important. Well, today, eye movements are very, very easy to measure. This is what's called a Tobii eye tracker. We have many of these in the lab. These are very, very simple devices that you just attach to a computer screen, a monitor, or a laptop, and very, very easy to calibrate, and it follows and tracks their eye movements very, very easily. So technically and cost-wise, it's a very, very easy manifestation or easy thing to do. The second thing I want to mention is that when you're studying eye movements, you're really seeing the brain at work. It's not just the eyes moving and deep thalamic nuclei that are making the eyes move. It's also the visual cortex interpreting that information. It's the parietal cortex that's figuring out where things are in space. It's frontal cortex making decisions. It's the frontal eye fields that are responsible for moving the eyes. In other words, visual search or recording eye movements taxes the brain at a very, very high level. So it really gets us a chance to kind of push the brain, if you will, to see what's going on. The second thing that I mentioned is alluding back to what I said, is that visual eye movements or eye tracking tells us something about how that person perceives the visual world as well. Nice study that was done by Drew and Jeremy Wolf here in Boston. What they noticed is they did a very simple study of looking at chest x-rays that had a tumor, and they asked novice radiologists to search for the brain tumor. And notice how this radiologist is like looking everywhere. Then they asked very experienced radiologists and attending to do exactly the same task. And notice how now the eye movements are much sharper, fewer, direct, getting straight to the target. Exactly the same task, exactly the same image, except here I haven't changed the question, I've changed the level of experience, right? So visual movements is not eye movements is not just looking where to look, it's also knowing where not to look. And the point is that visual search or eye movements can be learned, right? It's something that reflects the overall connectivity and development of the brain, right? So that's why we look at that. Here's the first virtual reality task that we developed. It's called the virtual toy box. We did a series of focus studies working with kids with CVI, parents, teachers, fellow clinicians, and so on, to get input as to what type of tasks are meaningful to these children that we would want to kind of probe. And the first one that we developed is called the virtual toy box. So what the individual does, looking here, you see the back of their head, they're looking at a monitor as if they're looking deep into a toy box with various toys. And there's the eye tracker down here. What they do is they have to choose a toy in isolation that they want to search for. It could be a blue truck, it could be a yellow duck, it could be an orange ball, for example. They have to choose the toy that they want to work with. Then what we do is we place it in a 5x5 matrix. So the number of toys never changes, but we change the number of unique distractor toys around. On low task demand, there's only 1 or 2 different toys around the target. On medium level, a little bit more, 4 to 6. On high distractor, almost all of them are different. So our theory is as we increase the task demands, as we increase the number of different toys in the toy box, the task should be harder, right? The other thing we do is called a color match task. We throw in another toy that's the same color, but not the same toy. Because we want to know, does the CVI child use the color cue to tell us that they can identify it, or is it really because they know the actual shape? And then the last one is kind of like throwing the kitchen sink. We add a very, very complex background clutter to really stress the system, if you will, to see if crowding and environmental clutter makes their performance worse as well. We scramble all these conditions, right, randomly. We show them to the child and we record their eye movements. And this is what it looks like when you do it. So here's a head-to-head comparison. On the left is a control. On the right is a child with CVI. The task is to find the blue truck. This yellow blob that you see here is where their eyes are. And just to keep in mind, they don't normally see this, right? They don't see anything on the screen except for the task. That yellow blob is just for recording purposes, so you can tell where they're looking, okay? Here's the head-to-head comparison as these 2 kids try to find the blue truck. And what do you notice? Do you see how the path is so much more complicated for the CVI child? Here's another example. Look at that. They finally get to it. They get to the blue truck, but look how much more it is. Here's the color match. Look at this. Check, 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 back to the blue truck. Control never does it. And the CVI task, in the maximal clutter, in this particular case, the CVI subject found it. And this one, they missed it completely, right? This is not something you would see just testing visual acuity or perimetry. The only way you would see this is to actually record their overall eye movements. They get to the target, there's no question, but much longer, much more complicated path. And this fits with the presentation that a lot of children tell us. They tell us that after a day of school, they're exhausted, right? Why? Their eyes are working much harder. Pound for pound, they can do the task, but they have to do so much more work in order to get there. Think about it. If I ask you to walk from point A to point B and you do it in a straight path, that's a lot easier than if you had to go zigzagging around trees and so on to finally get to the end destination. Compound that over hours and hours of classwork or a day in the world, no question that you're going to be exhausted, right? So the eyes tell us that they really seem to have more difficulties spatially localizing things in the environment. Now, I'm sure you're thinking, well, how do you know this isn't a basic eye movement problem? It's not. It really is a complexity issue. And this example here, this is the same child with CVI who just has to find the blue truck by itself. And what do you notice? They can get to it right away, right? When the blue truck is in isolation, there's no distractors in the environment, they get to it right away, right? You can't tell me that this is either a CVI or a neurotypical control. They look exactly the same, right, in this particular case. So it's not a baseline eye movement problem per se. It's a processing problem. When you really tax the brain, they then start struggling. They then start having more and more challenges to carry out otherwise a very simple task. And we can quantify this. We can put numbers to this actual experiment. What I'm showing you here is what's called a heat map. So what we do is we overlay all the runs on top of one of another so we can compare them. And this little dot that you see here is the error of whether or not the child was able to find the target. The smaller the circle, the better the child did at locating the target, right? So you can see in this particular case in our controls, under low task demands, you see that the circle is nice and tight. If we look under the high task demand, right, more toys, or more different toys, I should say, you notice that again, the circle is really, really tight, right? So as you increase task demand, these controls are the same. It doesn't change anything for them. Here's a child with ocular-based visual impairment. This is a child with ocular nystagmus related to ocular albinism. And what we notice is that at baseline, you see how their visual search pattern is a lot larger, right? Because the eyes are kind of shaking. Here it is at high task demand, looks pretty much the same in terms of area as well. So again, not really sensitive to high task demand. Our CVI, in contrast, on the low condition, somewhere in between. And as we increase task demand, now the area is much larger, right? So what differentiates CVI to ocular-based visual impairment and controls is the sensitivity to task demands. The more complicated the environment, the worse they do, even though it's the same task. And you can plot this in our groups. So we've tested about 15 kids on this particular scenario. And this is what this plot is here. On this axis is performance. The higher the number, the worse they're doing. It has to do with the area of that search area. So the higher they are, the worse they're doing. On this axis here is low, medium to high. This is task demand. So the more you move to the right, the harder the task is. Here it is in our controls, pretty much flat, right? Slight uptick from medium to high, but on the net, as a group, the controls are not very, very sensitive to task demand, right? Here it is in our ocular visually impaired. Very much the same sort of profile, but raised at a higher level, right? But overall, the sensitivity, like the lines are essentially parallel. Here it is in CVI. At low task demand, they're somewhere in between. And as you increase task demand, notice how much worse their performance is. And we think this slope, this increase, is an indication of just how functionally impaired that child is. The steeper that line, the more impaired they are to visual search and task demand. So we think it's an index of how sensitive they are to environmental clutter, to crowding, and so on. Another piece of information that's important, we have kids who have 20 to 25 visual acuity, so very, very good visual acuity, and their performance is essentially the same. So high visual acuity does not get you out of this predicament, right? So it's not just a question of like, oh, well, they're visually impaired, they have low visual acuity, that's why they're not doing so well. No, even in kids with CVI with high visual acuity or quote normal visual acuity, we see this same phenomenon. And this is important because when you take that child into a standard eye exam to go see the eye doctor, you're essentially testing them under low task demands, right? Reading an eye chart or clicking, lights on a screen is not a very visually demanding task. And they may do just fine, right? They may just do, they may be completely comparable to kids with neurotypical development. It's only when you tax the system, when you put them sort of in real world scenarios, that you see that the visual system breaks down, right? So again, it's this sensitivity to task demands that I think really is the hallmark sign of visual, or I should say, of cerebral visual impairment. We've gone further and we've coupled the visual toy box with wireless EEG recordings. You're probably all familiar with visual evoked potentials, right? Some kids of yours may get this. So a visual evoked potential is the recording of an amplitude, if you will, or a voltage change, or an increase of activity, if you will, in the visual cortex. This is actually one level higher. We don't just look in the visual cortex. We look at the entire brain to see how it's doing. So we have this EEG system connected or while they're viewing this visual toy box, And what we find in our controls, here's when the toy box appears. They get this initial sort of look, and then they move their eyes. So this is the initial period of when they're looking at the screen, and they have to decide where the target is. And what we see in our controls, you see at 300 milliseconds, 350, and so on, really, really robust activation in the occipital cortex here. Parietal areas, temporal areas. So the visual cortex is coming online, responding to this task. What does it look like in CVI? You notice the temporal cortex is looking pretty good. Dorsal cortex, not so much, right? Again, fitting with this idea of dorsal stream dysfunction, right? So the brain activity fits the wiring, fits the clinical complaint, all kind of coming together. That's the idea. You may be asking, well, that's all well and good, but are you going anywhere more in terms of this toy box and so on? Well, I think one thing that would be kind of interesting is to use the toy box as a way to probe what cues are particularly useful for that particular child. So for example, if I move the toy, does that enhance their performance? If I change the color of the toy, does that enhance their performance? If I change the lighting or the luminance, does that change their performance? If I remove the clutter background altogether, does that improve their performance? So this allows you to assess on a case-by-case basis what could be potentially useful as a visual cue for that particular child, and then translate that into the real world. And I'll give you an example of where we actually did that. Joyce, who's your friend? Hi there. Quick example to show you what we're doing with our sort of older adolescents and young adults with CVI. Instead of looking at a toy box, because again, we have to design environments that are the sort of behaviorally relevant to them, we created a virtual high school hallway. So what the person doing here is they have to pick a person they want to search for. It's a principal of a fictitious high school that's walking around while other people are walking around. And I'll just show you here. I'll play the video here. And the idea here, very similar to our case of the toy box, instead of changing shapes and colors, what we do is we change the number of people in the hallway. And what we find is, you notice that under low distractors, low number of people, both the control and the CVIs are able to lock onto the target and follow it fairly easily. If we put somebody who has the same facial features as the target, you see how the CVI has to go back and forth to check, right? And here it is under high distractor levels, lots of people, the control locks on, there she is, CVI late to find them as well. So very, very similar to what I showed you for the toy box. As you increase task demands, in this case, we don't change the number of toys in the background or different toys, we change the number of people in the environment, we see exactly the same relationship. So again, this is error as a function of task demands. Our controls essentially flat. Here's this again, steeping curve for the case of CVI. As more and more people are in the hallway, the harder it is for them to do the task. Again, fitting with the clinical profile with a lot of these adolescents with CVI tells us. The same task, the same individual, but if you make the environment more complicated, the performance starts decreasing. And again, fitting again with what we saw with the toy box. Here's an example where you can start using virtual reality, which I think in a more beneficial, for lack of a better term, or to probe actual function. This is one young man we've been working with CVI. Here's again the controls in blue, flat relationship. This is his performance on the hallway task. So again, he's much worse than the controls. And as task gets harder, his performance gets worse, right? So very, very similar. Slight downturn there, but on the net, he's very, very sensitive again to task demands. The first thing we did is we compared his performance when he was still watching the environment, and then also him moving through the environment. So we had to do the same task while he was moving in the environment. And what we found is his performance got worse. So even doing the same task and changing his perspective to move through the corridor, we made it even harder for him to do the task. So we know that this is useful because what we tell him to do now is, if you are in a busy place like the mall, the airport, or high school, and you're looking for a particular place, you need to stop, try to find your bearings, look where the target is, and if you don't find it, then you move on. And by doing this systematically, you have a better chance of perhaps finding the location you're looking for. Something, again, tailored to his needs. The second thing we did, which was kind of interesting, is we put the principal, the target, in a very bright yellow jacket, right? And what's interesting in this particular case, high saliency, we essentially normalized his performance. So he was performing at the same level as our neurotypical control kids. And let me show you a video that kind of shows you that. This is the same kid with CVI. On the left is using the yellow jacket, and on the right, the principal does not have the yellow jacket. Same individual. Finds it right away. He's missing it in this case. Another example. Again, finds it with the yellow jacket, still searching without. Right? So simply increasing the salience of the target, in this particular case, bright yellow, improved his performance tremendously, right? So sometimes the virtual reality is also useful to allow you to probe to see what type of cues might be very, very beneficial and useful for that particular child that you could technically or theoretically translate to the real world. I'll tell you a quick story, which is quite nice. The mother was quite surprised when she saw these results. And I remember she told me, "You must have cured my kid." And I said, "No, I didn't cure. All we did was just experiment a little bit in a controlled fashion to figure out what cues that were helpful and what things made it worse." And I think she really took this to heart, because we're very close to the families, as you might imagine. Well, I got a text a couple of summers ago where the mother said, we were on our way to a big amusement park in New Hampshire where there was a lot of people, and the family was feeling particularly stressed because the young man really, like I said, doesn't like crowds, and he gets lost and he gets really, really anxious. So the mother had a very, very clever idea to put the family in these bright orange fluorescent t-shirts. So there's the young man here. Those are the parents and his younger brothers. And apparently the mother told me they had a wonderful day, because not only did the young man not feel anxious, because at any given time he could find his family and locate them very quickly, the family also, by projection, saw that he was enjoying himself, was not anxious, and then themselves felt a lot less anxious as well. So just an example that sometimes very, very simple things can have a lot of impact, but what we need to do is figure it out on a case-by-case basis, because what works with one particular child with CVI doesn't necessarily work for another. And we have to also know which things are more impactful than others. Just to wrap up, something that I kind of want to share with you to kind of put this all into perspective. I had mentioned to you that visual perception is an iterative process, right? Information enters the brain, the cortex or the executives are constantly discussing it, sending it back down for more information to come back in, and the cycle churns and churns and churns and churns. That's visual perception in a nutshell. You can also break it down into two processes, what we call bottom up, which is saliency. That's information entering the eye into the brain, right? Bottom up information versus top down information. That's your expectations influencing the visual perception that you see. For example, the young woman versus the old lady, that's a top-down influence. The bottom up is the image. The top down has to do with your experiences and so on, influencing what you see. And when those two processes come together and align, that's perception. It's a combination of what's coming in and what you expect to see. What I suspect in the case of CVI is this. I think that bottom-up influence is impaired. It could be anything related to pathology at the level of the eye or information coming into the brain. And because of that, over time, that top-down influence, that visual repertoire doesn't build extensively, right? It's not anywhere as large as it is in the case of neurotypical development. And more often than not, there's a mismatch between the two. In other words, what's coming in to the child versus what they expect to see doesn't always jive. And I think that's one way to kind of conceptualize what CVI is. It's a mismatch between what's coming in and what they expect to see. And let me give you an anecdotal example of why I think this is the case. This is Matt Tejan. Some of you might know he does a lot of work with complexity. He's a teacher for the visually impaired in Connecticut. And he does a really, really nice study that we're working with him. And let me give you this example. This is a picture of a banana. This is a cartoon picture of a banana. This is a caricature of a banana. And this is an object that's shaped like a banana, has the same color of a banana, but clearly is not a banana, right? You have no problem distinguishing between these four objects. You know that they're fundamentally different and different representations. Why? Because your experience, your visual repertoire is very, very extensive, and you've built this over time. So what Matt was interested in, he was interested in figuring out, why do kids with CVI seem to not like things like comic books and cartoons, things that were a little bit more abstract, right? They seem to really have a hard time to understand or to grasp in terms of what they mean. And what he did is he came up with a study where he used a classic color photo, a realistic color illustration, the same object without color, and then an abstract color representation of the same object. And what he finds is that his age-matched controls can distinguish all four of these, but his CVI kids are somewhere in between. They have a really hard time that as the objects become more and more abstract and less information, they have a harder and harder time identifying what that object is. And let me give you an anecdotal example that kind of drives this point home. So we did this study with him. We gave this sort of a randomized way. We show this to all the subjects, and we just kind of go through like a flip book, and the child has to record what they see. So in this particular example, we showed him this caricature picture of an elephant, right? So we go through, the child looks at it, and they say, okay, so what do you see? He says, remote. We're like, okay. So we move on, we go through the other ones and so on. Then after we finish the test, we go back and we ask the children to explain their choices for the ones that they got wrong. So we go back to this picture and we ask him, so what did you say this was? He goes, it's a remote, obviously. You're like, really? Why do you say this is a remote? Does anybody have a Sony PlayStation at home? That's what a Sony PlayStation remote looks like. So there are the two eyes, there are the ears, right? That is what he thinks that object is. That's his visual repertoire, right? He spends a lot of time playing video games. This is in his close vision, right? So he has this very, very close. He probably has this at sort of a decent level of detail. He thinks that when he sees this abstract image, he is going to pair it with his visual repertoire of something that's very familiar to him. That's what I mean by this mismatch. What's coming in versus what he expects to see doesn't jive, right? That's what I mean by that mismatch. Okay, last slides. The laws of neuroplasticity, things to think about and summary. First of all, plasticity is a fact of life, right? This is going to happen. The joke is death, taxes, neuroplasticity. It's going to happen. It's an intrinsic property of the brain. It can be compensatory and adaptive, as I showed you in the case of the young man who was reading Braille, for example, with his lips, but it can also be maladaptive, right? There are examples of maladaptive plasticity as well. How the brain wires isn't always a beneficial thing, right? Timing matters. This has repercussions for rehabilitation. So for example, an individual who has neurotypical development and has a stroke and has a visual impairment, the way that we rehabilitate that individual may be very, very different than for an individual who's born with a brain injury, right? Because we're not dealing with the same type of brain, a different type of wiring, a different type of machinery. And we have to take that into consideration. You use all your brain all the time. It's not 10%, it's not 15%. That's just some cocktail party myth that's been spread around. You use all your brain all the time. And the key is to learn how to promote that, how to guide that plasticity in the most beneficial way possible in order to optimize development. And that's my last slide about how can we enhance plasticity and brain development. The first thing I say to parents, whether it's ocular visual impairment or CVI or anything, who ask me, what should I buy for my kid to help them? I say, buy a Nike t-shirt. Just do it. Get out there. Interact with the environment as much as possible. And you'll see how I'm going to drive this point home in just a second. The first point I like to say is what's called love learning, right? That's very, very important. Just kind of prepare this sort of growth mindset, promote discovery as much as possible, especially during those critical period years. Really kind of encourage the child as much as possible to interact with their environment. We, as parents, we tend to really influence more, or we rather tend to decide more on results rather than the process. Think a little bit more about exploration. It's okay to make mistakes. It's okay to discover new things, and less about being successful at a few things. So think more about the process rather than the results. Balance repetition with novelty. There's two ways that the brain likes to learn. It likes to repeat. If you try to remember a phone number, you have to rehearse it. The brain likes that. But after a while, seeing the same thing after a while, it gets bored. So sometimes novelty is the better way to enhance performance as well. And I think as parents and teachers and clinicians, we need to be able to use repetition and novelty in ways to try to enhance learning as much as possible. So we have to be able to go between those two strategies. And finally, what I'll say with here is get comfortable with the fact that success comes with effort. I see a lot of children, even like I said, with neurotypical development, who say, you know what, this just isn't for me. You know, I just can't do this. I'm not wired for this. I'm not built for this. That's just simply not true. Get used to the fact that success comes with effort, because that is the brain wiring, practicing, repeating, trying to form those connections in order to develop those skills. Secondly, what I call buffet style learning, right? Concentrate on breadth versus depth of skill development. You know, this is classic in kids, for example, who have a particular gifted athlete, right? Let's say the kid is very, very good at baseball. That's all they do all day. They don't play any other sports. They don't do any other thing. They are 110% dedicated to actually that particular craft or that particular skill. I would argue that that's not necessarily a good thing from a brain development standpoint. The more you can expose the child to different skills, the more overlap you create, the more problem-solving skills, creates this idea of well-rounded, adaptive, creative, flexible thinking. A great documentary I saw about Wayne Gretzky, the famous Canadian hockey player, he said, yeah, I'm a good hockey player, but that's not the only thing I did. I played baseball in the spring. We played football in the fall. We did all sorts of things. And he was convinced that by being good at lots of things, that made him an excellent hockey player. Not just, and he even argued, if I was born today and my parents forced me to just play hockey, I probably wouldn't be anywhere as good as I was. And I think that's an important takeaway from that, that breadth over depth is actually a good thing. Third, emotional learning. This gets back to that earlier question, which I think is very, very important. Understand the link between decisions and emotion. Emotion is the older brain. It's kind of how the brain figured things out. You know, pain, no pain, love, not love, hungry, not hungry, the strong emotional connection. But what we did as primates and as humans is add this layer of complexity called the cortex, which was kind of figuring out and trying to rationalize those emotions. So promote that link, allow those links to strengthen, particularly for child development. Understanding or rationalizing our emotional reactions is, I think, a very positive thing for development as well. So learn the link between emotion as well as thought. And last, I said the Nike t-shirt, get busy. I think that's really, really the important thing. I think as parents who have children who are visually impaired or have disabilities, we tend to kind of be protective. We're afraid that they'll hurt themselves. We're afraid that that they'll get frustrated or so on. I think we really need to promote and let them explore the world, engage with the world. We know that there's a lot of scientific evidence about the benefits of environmental enrichment, right? Children and animals that grow in very environmental, complex, and enriched environments have brains that thrive. This was a great study by a group in Granada that showed that children who had extensive not physical therapy, but recess, right, time exercising, showed really dramatic changes in overall gray matter in parts of the brain responsible for language, right? They weren't training language per se. It was simply good cardiovascular exercise that promoted these changes at the level of the brain. So develop an inner mind that is exploring the outside world. Remember, that's what the brain does. It tries to create an inner model of the world so it can interact with the outside world. Set up for success, create environments where that child can thrive, and really reinforce this idea that success comes with effort, and be cautious to not frustrate the child either, because that has obviously a negative effect. And finally, it's not over in terms of the critical period. It may be harder. It's a lot easier to learn a language when you're 6 and not 60, but it's still possible. But as you get older, you need to be that much more creative, that much more intense, that much more repetitive, et cetera, to get those same skills in there. So hopefully those 4 points are positive takeaways, not just for CVI, but just for development in general. And again, not just children, but for all of us, because I think a lot of these ideas are just sort of grounded in what the neuroscience tells us. And I think that is it. So we're half hour over, yeah?
It works for me. It was amazing.
Okay, good.
Do you have time to answer any of these
Absolutely,
questions?
absolutely. I am totally fine. Just a little bit of water. I'm all yours.
Sorry. Sherry Lowe is asking how to get other professionals delivering special ed services to consider CVI referral.
Yeah, I'm sorry, is this specifically on the clinical side or on the education side?
Is Sherry still here? Does she just want to ask her question? I'm not sure if she's still here. I'm sorry.
It's okay. I think there's a couple of ways to answer it. I can sort of share some thoughts.
It does say special ed providers.
It is special ed providers. Okay. So I can't really speak that much on the education side, except for the fact to say, so basically what you're saying is you have a parents, for example, who have a child with, say, CVI or visual impairment, and those on the education side are not understanding that certain accommodations need to be made, certain strategies need to be incorporated. I'm guessing that's the question.
Sorry, I just found my unmute.
Okay, no problem, Sherry. Please go ahead.
No, I kind of gave a case scenario on there, and I'm like not trying to self-diagnose, but
no, that's fine. Go ahead.
But it has to do with all these things you kind of made reference to as far as developmental age. My granddaughter is 12, and she's going into 6th grade, so she's now beyond that critical period, obviously. But she has been in special ed, I don't know, I think since 2nd grade, and just in the last 2 years, they thought, well, let's get her some glasses because she struggles with language. But she's fatigued. She's kind of locked herself into this one area, which, not getting out of environmental enrichment and her art. She seemed to be a left brain learner, so to speak.
Right.
But the parents both were special ed students in language disabilities as well. So the other part of that question was, how genetically based can this possibly be? And should I don't say much to them, but I'd love to be there at the special ed meetings and say, what else can we do? What are you guys simply looking at, and what else can we really should be looking at?
Yeah, absolutely. And again, I'm, a lot of stuff, intertangled in there. And I think it's a great example of the challenge of dealing with these kids, because there is so much going on, so many barriers as well, particularly again on the clinical and educational side when you're working with people who just don't get it, and that's a lot of the fight. There's no question. I think in this particular case, if I can speak in sort of general terms, not just I think you mentioned this was your granddaughter, is that correct?
Yes.
Yeah. So maybe just in general terms, what I've seen in my experience is that very often on the education side, they tend to listen when the parents come with weight. What does that weight look like? Typically, it could be a letter from an eye doctor, for example, or a physician who has examined that child and says, yes, this is what I think the visual difficulties are. Typically, it's also good if you can work with a provider, an eye doctor, for example, who knows the educational needs and demands and talk about those visual needs and deficits as a function of how that child is in the classroom. That I find has a lot of weight, so that when the family goes to the IEP meeting and there's, let's say, some pushback from the teachers that, oh, this kid, is fine, or what we're doing doesn't need to change, it forces them to rethink their model. So I would say the first thing I would say to all parents is find providers who are allies. That you could go back to them and say, look, the teachers don't see this particular deficit, or they don't think this is a particular issue. Do you think that's a particular issue? And can you be that explicit in a letter or a phone call or something along those lines? And I find that more often than not, the schools will bend if a person of, quote, authority speaks to that effect. So the first thing I would say to all parents is find a provider who's going to be an ally, who's going to be an advocate for your child. And it's not just that eye exam and prescribing glasses. It's going beyond that. It's really all their needs, their visual perceptual issues. The second thing I would say is that if you do indeed see so much pushback from the educational community, it might be time to think of maybe switching that educational community and services as well. Some institutions are going to be much more amenable and more open to these type of needs and adjustment that the child has. So I think it's a way to kind of diagnose, if you will, if whether or not it's the appropriate learning environment for that child, depending on how much pushback they give. Is that helpful at all, Sherry?
Yes, it is. But I'm like, what would be the other institutions to consider? if for a better and different environment, learning environment is one. And then number two, I'm a grandparent, so it's like getting my daughter to, even
to listen. Yeah.
to be that assertive to say, let's get another physician's opinion or something like that.
I can't really comment, when you're sort of one level removed, if you will. I can certainly see it from the parent standpoint when they come to us, or the primary caregivers of the child. But I think someone yourself was in a unique situation of sort of having that bird's eye view, or that 1,000-mile in the air view. I think you can come to them and say, look, from my experience seeing lots of kids like my granddaughter, these things may help push things in a particular direction, and you can maybe steer that dialogue towards another provider that could maybe more in line to what the child needs, more in line, or steer that child to an education system that may be more in tune. You've got sort of two barriers. One is what the child needs and support, and also obviously your daughter, to convince her that this needs to be done.
Are you going to include at the end of your presentation? I would love to get some referrals of people that.
Sure.
Would be great for us to be able to refer families to.
Absolutely. I'm happy to provide, providers that we work with. We have a great working relationship. Terry Schwartz is one pediatric ophthalmologist who leads the pediatric ophthalmology at Cincinnati Children's Hospital. So Ohio, not too far, but certainly in your realm. And again, I go back to this point that really it's about aligning yourself with like-minded people on the clinical side, on the education side, on the advocacy side. If you're working with people, I don't care what their degrees are and what the letters are after their name and where they work. If they don't get this, they're not going to help you. So it really is a question of working and identifying people who are like-minded. I would say that's extremely important.
Thank you very much.
Certainly. I hope that's helpful.
Hey, Pam, I think 3 of the questions in the chat were yours. If you want to ask the most pressing ones, you're up.
Sorry. Well, I had a question about so if I had a student who had CVI and also had a refractive error.
Yeah.
I've had eye doctors who have been hesitant to prescribe correction. And, in my mind, it makes more sense for the brain to try to process an image that's clear versus an image that's blurry. Are there clinical reasons that they might want to wait? I mean, the reasons that I've been given is that they wanted to have more consistent looking behaviors prior to that correction. But I just didn't know, what would be the virtues of waiting versus going ahead and prescribing?
Absolutely. Great question. And to be perfectly honest with you, it's something that the ophthalmic and optometric communities are struggling with. There are practicing guidelines that have been laid out for pediatric vision in terms of how much correction, when to give, when to scale, and so on. But they were never designed for the CVI population. So that's an important thing to think about. So there's nothing that says that those rules that have been ingrained for the past 10, 15 years are hard and fast rules for the case of CVI. But I can think of scenarios where not necessarily giving the full correction right away could be warranted. One example is if the correction is very high. So for example, if the correction has a lot of astigmatism, a lot of cylinder, right? If any of you have astigmatism and maybe think back to the first time you got those glasses, you're very, very kind of woozy, you get headaches, all sorts of things. That child isn't necessarily going to report that to you. As an adult, maybe you'll say, yeah, this is a little too much. I kind of see the doors on a skew, oblique, but a child may not necessarily report that and just reject the glasses. So there may make sense that when there's a high correction, particularly astigmatism, to kind of ease your way into full correction. So that's one example. So basically I think what the doctor is saying is, I want to ease into it to kind of break the glasses in, if you will, or break the child into the correction, or at least see that the child doesn't reject the glasses. So if I just take that leap to full correction, the child may just reject them, and then I've kind of lost that window completely, right? It's very hard to walk that back. So sometimes easing your way in is worthy. The second thing that you may say is that if the visual acuity is so poor and not correctable, Let's say that even after best correction, you can only get to say 20/60 or so on. So that high correction doesn't necessarily give you that much change in acuity. You may say to yourself, well, if they're nearsighted, it's probably pretty clear without correction up close anyway. So you say, okay, in the early ages, that child's world is pretty much up close anyway. I'll think about holding off on that far correction, right? That nearsighted correction for distance a little bit later as that child's world, so to speak, expands. That could be a rationale as well. There is also a case, it hasn't happened often, but I think is certainly worthy of mentioning, is particularly in nearsighted children, if you give them that full correction, some children are overwhelmed with the clarity, right? They're like, wow, everything is crystal clear. And all of a sudden, those issues of complexity and clutter and being overwhelmed, all of a sudden creates that rejection and essentially makes the child, quote, could make the situation worse. The problem is we're talking about a patient that doesn't tell us, I don't know a 3-year-old or a 2-year-old that puts the glasses on. I'm like, oh no, that's way too much clutter, and that's the problem. So a lot of it, unfortunately, is based on clinical judgment. There are no hard and fast rules with this sort of thing. It has to do with the degree and the type of correction. It has to do with how much that correction buys you in terms of acuity. It also has to do with how well that child tolerates that correction right away. But what I would say is in these particular situations, you really have to have a rapport with the eye doctor to know that this is not a one and done sort of situation, that it'll probably take some refinement, over 6 months, a year, to try to get at the prescription that is optimal for that child.
Thank you.
Okay.
The only other question that I just wanted to ask is, we typically teach our students the systematic scanning process from left to right and top to bottom.
Yeah.
And, for students with an ocular impairment, it tends to typically improve.
Mm-hmm.
So with students with CVI, when you said that they were having difficulty with that, will it improve, or will they always have these difficulties that we just need to accommodate for, like with color or?
Yeah, great question. I think what I would say the big difference from what we see, and again, this is speculation on my part. I don't have hard evidence for this, but what we think is happening is that in the case of ocular visual impairment, you can kind of think of it as a spotlight, right? Where they need this sort of systematic scanning to help stitch that picture in their mind, right? Okay, so they're sampling the visual world in a systematic way to allow them to build that stitching in their mind of what the visual world is. The problem in the case of CVI, that's not to say that strategy wouldn't work, but I think the reason why it doesn't work as well is because there's also a spatial processing problem. Their ability to understand the relative position of things in space is also impaired, right? So there's a double layer of difficulty. I know that, for example, Gordon Dutton out of Scotland doesn't use this sort of linear approach. He uses something called wagon wheel, where they go from center out and then back, out and then back, out and back. And this idea that every time you come back to home base anchors that spatial registration of you to the visual world and where you are in the visual world. That's the rationale, right? But I think this is a great example of how things that we would think would work, or we know work in the case of ocular impairment, don't necessarily translate in the case of CVI, because we're not really dealing with necessarily identical problems. We're dealing with different machinery, right? So I think we have to adapt accordingly. But that's a great question. So I guess the only thing I would say from this is, just because something is tried and true for ocular-based impairment, you can't assume that's going to work as well, if at all, in the case of CVI, because it's really a different brain.
Thank you.
Sure, great questions.
Kevin Kellerkern is asking, you had mentioned earlier that you would tell us why it was important or interesting that the brain processes the image upside down in the visual cortex. Did I miss the explanation?
I'm not sure if I said that, then I misspoke. I think it's interesting that the brain is wired that way, but I don't have an answer as to why the brain decided to do right and left and right, left and right, up is down, down is up. Oh, I'm sorry. I do know what he's talking about. I apologize. Great question. Sorry. Kevin, is that right?
Yes.
Thank you for being on the ball. You are 100% correct. I just sort of flashed into what exactly you were referring to. Okay. Let me just find the figure in question. So let's just play this video again. Can everybody see this? This is the video there. Okay, so I'm just going to play this. It's not the best figure, but I think it'll help make the point. You'll notice that the dorsal part of the brain seems to be more impaired than the ventral part of the brain, right? Right below this, not obvious obviously on my picture, are the optic radiations, right? So the optic radiations, you can imagine the eye is out here, the thalamus is deep in here, and then the optic radiations go back to the visual cortex, right? They're not illustrated here, so you have to kind of use your imagination. The superior bank of the optic radiations, which are closest to this site of injury, are responsible for the inferior visual field. The superior visual field are related to the inferior bank, which are largely intact because they're close to the temporal side. So we think the reason why CVI is normally or typically associated with an inferior field cut is because the superior bank of the optic radiations are right up close to the parietal cortex, where most of the damage is in the dorsal stream. So there's an anatomical correlation to why it's typically an inferior field deficit. It's not the best figure. I should show you a better sort of anatomical one, but that's the rationale. Good, good, very, very good question. It takes some more figures to explain that, but that's what I was getting. So it wasn't so much up is down, down is up. It's just why there's an inferior field cut actually fits anatomically to where the damage is, or more typically, I should say, where the damage is.
Do you have time for one more question?
I'm not going anywhere.
Okay. Jennifer Katz is asking, with Hardy, is this a test that is everywhere or only at certain locations? How can we help advocate for our families to look into this test, and would it be beneficial?
Yeah, great question. I get this quite a bit because every time I show this, I see parents are like, I want my kid to get that, you know, sort of thing. But the question, because there's two sort of points. One is it's a technique. You can, in principle, do this with any MRI scanner. It's just a question of setting up the parameters in order to do that. But that's not the hard part. The hard part is the reconstruction. And fortunately, I've got people in my lab a lot smarter than I am who know how to do this. It's not a typical clinical scan per se. It's a technique that's being developed for many. It was actually originally developed. One of the first implications was for traumatic brain injury. And there was some very, very interesting work being done with that. And it's interesting because the results were very, very subtle for TBI. And I remember watching that lecture. I said, you know what? We need to do that for CVI. And then lo and behold, that's when we revealed these big changes. So it allows us to look at the brain connectivity, which I think is good from a research perspective. But it then brings me to my second point is, what do you do after this, right? Because at the end of the day, I think the diagnosis of CVI is going to be largely based on history and behavior, not so much the imaging. What happens if you have a child, for example, who doesn't show this dorsal impairment, for example? Does that kid no longer have CVI? So you have to be kind of careful what you wish for. At the time being, I think Hardy and these advanced neuroimaging techniques really are in the research phase to help us understand the neurophysiology of CVI, to better understand the deficits that they have and how they relate to visual problems in general. But I don't think it's prime time yet to say that every child with suspected CVI needs a Hardy in order to demonstrate, see, I told you this kid has CVI. Chances are you're going to be able to figure that out anyway without all these sophisticated mechanisms. Who knows? Maybe in the future it becomes something much more open and you know, readily processed. But for right now, I still think it's more in the explanatory phase or research phase than it is for clinical diagnosis.